Point one. Okay, thanks.
Welcome back to Greenpeace Deep Arctic Expedition. I'm right now in our live studio, so behind me you can see how we control all the cameras, and this is from yesterday's dive at a place called Loki's Castle. It's a hydrothermal vent field where volcanic hot springs emit this hot fluid up to 300 degrees. And this might be Loki himself. Look at that guy. Loki was a Norse god that named this place, and well, in fact, many of you will probably know Loki better from the Marvel movies. And at the very end of our dive, we've got this beautiful visitor. It's a Dumbo octopus dancing into frame. And if you want to have a look at this dive and any of the previous ones, go into Greenpeace International's YouTube channel, where all of them are. And now we're going in for the next dive. It's something totally different. It's called Schulz Bank. It's a seamount going up from 2,500 meters up to 600. And this is covered with life. Deep sea sponges, soft corals, many, many other things. You don't want to miss this. Let's go live.
Wait here until we are live. It's good, or do you want to proceed?
I mean, we can start
our
because
we can start
yeah.
our dive here. No, shall we zoom in? Okay.
If you want. I don't know if they've started yet.
Me neither.
They will tell us, I hope.
So we're here on the Schulz Bank
Yes, we're on the Schulz Bank.
in 1,090 meter depth. Let's start flying, please. Yeah. Okay.
So we're ascending the Schulz Bank Seamount on the west of the Mid-Atlantic Ridge in the Arctic. This is a huge mountain, and this is probably the best studied mountain in the area, because people have been coming to this mountain since 2008, since the discovery of Loki's Castle. And the reason for that, of course, is its proximity with Loki's Castle, and the Norwegian expedition at that time wanted to explore mountains in the same area, and especially mountains and sponges. So Schulz Bank is famous for its colorful and extremely rich sponge grounds. We are currently at 1,087 meters depth, and we're ascending this Schulz Bank seamount.
So far we mainly see anemones, so there are no sponges on these pictures.
So
There's an urchin.
currently in the shack, my name is Paco Cardenas from the Museum of Evolution at Uppsala University in Uppsala in Sweden, and
I'm Anna-Nina Lortz from Hamburg. When we zoom in now, thank you, we'll be able to see brittle stars.
so you can see two laser points. These are 20 centimeters apart. This gives you an idea of the scale of what we're looking at.
And we see lots of brittle. Exactly, we see lots of brittle stars. They are very abundant here.
There seems to be little tubes
Yeah.
on the floor.
Yes,
Could these be
Yes.
like worm tubes? Or?
yes, but you see this little white brittle star moving to the right,
Oh yeah,
just above
the red ones.
yeah, the dominant species is Caternials,
Trying to escape.
and they can occur in huge numbers, like 1,000 per meter square.
There's also a few shells here and there.
Yes.
So we are here at 1,083 meter depth in our dive number 9 of the Deep Arctic expedition. We left Ireland the 8th of May. So
We may
it's been a while.
fly on, thank you. And this is the 1,001st dive of this vehicle.
Absolutely.
Amazing.
Mm. So this robot is connected to our boat, our research vessel, through a cable. And we are currently in a container that is on the deck of the boat. And we have before us many different screens that not only show us the main camera stream, what you're also looking at, but also a lot of different angles, points of view around the robot, so that the pilot has a better grasp of where the robot is and what's under it, on the sides, and so on.
Is this a sponge coming up?
There's a large, more,
I'm pretty sure it is. This yellow thing and with a
It's
feather
possible.
star on top?
yeah. And the little yellow things on top are other sponges. So it's sponges sitting on sponges. And the large sponge is probably a geodia. It's going to be one of the common species here on this mountain.
Can you please look at
Yep.
anemone and off?
There was a crinoid on top.
Yes. And more ophiurids. Camouflaged. Can only see this pink anemones pointing out.
So Anna-Nina and I are scientists, and we're logging our observation so that when people will look again at this video, they can use it for science, to study communities in the deep sea, eventually also for description of animals and behavior. And Annenina also can actually move the camera and zoom, and this is quite a luxury for us to be able to do that.
So many girdle stars.
Yeah.
Girdle stars are quite funny. They have a big influence on the seafloor, and there's so many because they move quite a lot, so they turn over the top layer of the sediment. When they lose an arm, they can regrow it, wiggle along.
So these stars feed in the sediment. They don't feed in the water column.
No, they feed on the sediment.
On the sediment, yeah.
So their mouth is in the middle, but on the lower side of the central disc. So they have a central disc and then 5 arms, and they're fast.
So do they eat like detritus, or do they actually eat animals that live in the sand, kind of?
I guess both. Because there must be enough nutrition here to support such large populations.
So yesterday we were not far away. We were at Loki's Castle, which was a pretty amazing dive.
What's Loki's Castle?
Loki's Castle is where hydrothermal vents are in the Arctic Atlantic Ridge. So these are the most famous hydrothermal vents in the area. And it's during the expedition that discovered these hydrothermal vents that researchers also kind of explored the Schulz Bank for the first time. And that was July 2008. I was part of that expedition. I was a PhD student at the University of Bergen at that time. So I've been to Schulz Bank before. But that was a long time ago. So I'm very eager now to see.
Yeah, we get a best move there on bearing.
To explore new parts of this mountain.
340, continuous, 0.2 please.
340, continuous at 0.2.
Please, yeah, thank you. Bring him in a bit and then back out. So you'll be headed just like that.
So this mountain is more than 2,000 meters high, but it's under the water. So it starts at minus 3,000 pretty much, and the top of the mountain is at minus 600.
And it's very steep at the moment.
At the moment it seems very steep, yes.
I would love to zoom in once again.
Okay, do you want to stop?
That would be great. Thank you. Thank you. Once your eye gets used to these girdle stars, you can see lots. There are more than 10 here on this image.
Yeah, there's one crawling, a very funny way of walking, of moving.
Yes.
Ooh, there's one of your animals.
Yeah, you're getting very good at spotting them too. That was an amphibot flying by.
So Annenina is a carcinologist, is that the word?
Yeah.
Yeah? Meaning that you study crustaceans, but more specifically amphipods, which is a huge. How many species already?
10,000.
10,000 species
Of
worldwide.
Yes, worldwide of amphipods. How about sponges?
Sponges is also 10,000, nearly, we're getting there, nearly 10,000 species.
Sorry, we're flying on, please. Okay. And what, what are sponge people called? Like,
we don't really,
taxonomists of sponges?
we don't have a fancy name like you, I think. People call us sponge biologists or spongiologists, but it's a bit a mouthful, so I don't know if people use that a lot. Yeah, sponge biologists is fine. Okay. We are several zoologists on board, actually, so with essentially expertise in sponges, deep sea sponges, and deep sea crustaceans. But some of us, like Sergi Taboada from the museum in Madrid, or Anne-Helene Thunberg, or you also have expertise in other groups. I have to say that I'm very focused on sponges, so I'm not so good with other groups of animals, but I think we cover quite a lot of knowledge, all of us on board. Here we are at 1,066 meters. I am logging the depth now and again, even though the ROV is measuring the depth at all times, and we will have that data at the end of the dive, of course.
And even though we have a few experts on board, of course we're all working in our international teams and with our peers.
Yep, yeah. And usually working at a museum gives you access to many different experts on many different groups. Because we're both working at the museum.
Yes, I recently started at Senckenberg Museum.
Where is that museum?
The big museum is in Frankfurt, but I'm based in Hamburg, a little smaller outpost.
Mm-hmm. And I'm at the Museum of Evolution, which is is a part of Uppsala University. And actually, today is a very special day for us, Uppsala University or Museum of Evolution, but I would say for all taxonomists, because the 23rd of May, today, is the birthday of Carl von Linnaeus, probably the most famous Swedish scientist ever. Yeah, and maybe while we're ascending, I can say a few words.
Of course.
Linnaeus is a Swedish scientist born in Småland, but did his whole career in Uppsala, at Uppsala University. He was a professor in 1743, so this is 18th century. And he completely revolutionized biology by giving scientific names to every living organisms that crossed his path, which is thousands and thousands of animals and plants and fungi, all types of living organisms. And since then, all scientists and biologists have followed his discovery or his achievement, trying to describe and name all living organisms on our planet. And we are still doing that today, you and me.
Didn't he think that sponges were plants at the beginning?
Yeah, so that's, yeah. Of course, Linnaeus, during the 18th century, many things were unknown or poorly known. Is it this?
Yeah. There's a sea star coming up.
But yes, Linnaeus thought at the beginning that sponges were plants. But later on in his career, he moved them to animals.
Yeah, but I heard that you were looking for types and then they were found in the herbarium. I thought it was quite interesting.
Yes, yes. He had some specimens of sponges in his herbarium. That is true. Tell me when you want to stop as well. But it is.
It's quite
And
steep.
Andrea, tell us also if
can we
it's.
stop here?
Yeah,
Just
yeah,
for a
yeah.
second.
Of course.
I'm trying to look into these burrows. You see these little holes there, the
Yeah.
dark ones?
Yeah.
Well, I assume that a gigantic amphipod builds them. It's called Neohela.
Okay, right, slow to point 1.
So when we settle with the ROV,
Okay, bridge, all stop.
ROV means remote operated vehicle. It's fancy name for the robot. It always creates a bit of dust and sand,
Think you got that, Baz? Just come back to me.
and we usually wait for it to clear away.
Thank you.
And it's kind of tricky when we stop because we also need to tell the ship that we stopped, because of course we're hanging. The robot is hanging by a cable, so the ship and the robot need to work hand in hand and follow each other.
Then I get dinner.
And now we're focusing on this little hole just, or is the burrow
Yeah,
just in front of us.
yeah.
What I assume it could be built by an amphipod
Mm-hmm.
from Neohela. But I can't see it coming out. No, maybe it's not. No.
Could you, after, focus on the sponge?
On which one first?
On the white one.
Okay.
I'll try and center it a
Yeah.
bit more.
A little bit more, yeah.
Both are-
That would perfect.
What's on it?
What's on it? Brittle star, no? So here you see several sponges. You see the white branched one, which could be Lysodendrorix complicata. And then we have another rock on the right with an anemone and another sponge next to it, and that's probably Tentorium semisuberitis. So both of these are quite well-known sponges. And the little yellow one on the left, we don't have a name for it. We know it might be Aplysia, but we've collected the sample a few dives back, and we need to bring it to lab and do some DNA studies to be able to give it a name. Are we in a position to sample here at all? Are we stable? The Lysodendrorix, because
What do
we
you want?
the
This-
white one,
Okay.
because this
So
would
it's this
be
one.
yeah,
Yeah.
exactly.
Should be able to, yeah.
What? How do you want it?
So slurp would be fine.
Scoop. Slurp?
Scoop.
What do you think?
If scoop is helpful for you to catch something else, I'm not against it.
You prefer slurp?
But otherwise, flurp will be much quicker and yeah.
Let's look.
Okay, then give that a go. So let's look at
So this little white sponge is very important to us because we are trying to study population genetics in the deep sea, which is very challenging, of course, because you need a certain number of specimens to be able to do population genetics. And here, of course, these places like the Schulz Bank are very difficult to access and demand a lot of logistics and lots of equipment and so on. So when we are on site, we try to collect We focus on one species and we try to collect several individuals. Why do we want to study population genetics? Because we want to know how the species are connected in the deep sea. We want to know if populations are isolated, maybe on different seamounts, or if they actually reproduce with other populations on the next seamount, or on the other side of the mid-Atlantic ridge, or with populations nearer to Greenland, etc., etc. So that's why we are now subsampling this white sponge.
Until it's done with your next week. The other way. The other way. Down. We're using the left arm, Anna.
Oh, yeah, sorry, I was focused on the completely wrong sponge.
Oh,
Sorry about
me
that.
too. I hadn't seen that one.
Yeah, that was outside.
This one.
But it's
No,
a, it's the
not-
same.
it's the same-
It's
it's
the
the
same
same one.
species. So
It was just outside of our
Yeah,
monitor. Sorry, we didn't have that one.
I thought it was this
no.
one.
No, all good.
But then
It's the same.
can I just make a quick zoom then?
Yeah, I just ignored your picture.
Sorry.
We go down a bit, then we have the Ascidian and the Anemone.
Sure.
Honey.
Oop, there you go. Yeah, it's the same species. It's a very nice setting here. We have the same species, Locedendorix. Next to it, what seems to be a small bryozoa, brittle stars, anemones on the left,
And this blob
tunicates
on the
as well.
And the pink blob on the right side is also an anemone.
Yeah, probably
It's-
closed.
Yeah,
It seems
that's
that
what
it.
it's on.
Yeah.
Just, it's feeding.
Yeah, and the big potato-shaped on the right is
Left.
an
You mean
Ascidian. On the left, sorry, yes. On the left is an Ascidian. So we have a nice little garden, yes. But the one we're interested in is really the white.
Oh, now the
Fluffy.
a crustacean was missing. Now it swam.
Oh, yes, I saw it.
Yeah, green.
Oh, and look at this one.
Oh, yes,
Maybe we could slurp it
exactly.
together. You see this little tiny one?
This one.
Yeah.
Yes.
So,
I'll take that one first.
okay. We're going to use a
Can I
sort
go?
of.
Can I start?
Yes,
Yes,
please.
Okay. Just zoom it out a little.
Patty is our
Okay. Zoom back in so I can see.
is moving the arm from.
This is our big, go ahead.
Sample C star after the sponge.
Okay,
Okay.
we copy that, thank you.
Okay, so just walk me in where this little first little crustacean you want.
You're right above it.
So it's just
Here.
that.
Yeah.
Yes.
Please,
Oh yeah.
Tell me when you're ready.
Are you good in that area, guys?
Yes.
I think we're good.
You're right. Good. Yes, please.
A little bit down.
A little bit down.
I think it's in.
Okay.
Stop.
Okay, stop.
So it
Thank
sort of
you.
works like a vacuum cleaner.
Okay, so
Now we'll go to the sponge just behind it.
Same tank, right?
Same tank.
Yes, they can be in the same reservoir.
Do you want me to zoom in?
No, I just want to make sure I get the right white one.
The little fluffy one.
yeah.
Yep.
Little zoom. Okay.
Select.
No, not yet.
Yeah,
Okay, just go.
no,
Perfect.
That's it, yeah.
Perfect.
okay, off.
And we went to the starfish with that little out there.
This
yeah,
one?
that's a brittle star.
Brittle
No,
star?
We actually
That's it,
no. We just got a request from.
yeah. Jenny.
It's up here
Ah, it's
on the
this
right.
other one.
I'm pretty sure
Okay.
that
Yeah.
they mean this
Yeah.
this one because we haven't had that one.
I'm going to see if I can reach from here. Should be able to, yeah.
Yeah.
So
What do they say?
They say great job, Patty.
Thank you.
I think this is Hemonaster. This is what Jenny is
Little zoom there,
communicating to me.
Oh, yeah?
So I will write that we are sampling.
I can warn you because I know they produce quite a lot of slime.
Okay.
So
Then could we put it in a different slurp?
Yeah.
Yeah.
We should keep it separated because if they produce a lot of slime, this
do.
will contaminate our DNA for of the sponge. We don't want
exactly.
Okay.
to have that. Should we? Do you want me to write, or
It's all the same cast.
Okay.
it's all the same
Slurping.
cast but in different slurps?
Keep it on, keep it on.
Oh, excuse me. Was it?
Yeah, that's it.
What am I doing?
That's okay.
Okay.
No, I just touched this one.
Finish sampling?
Yes.
So this one is went to slurp 3? Is that
Two.
correct?
Two.
Two. Okay.
So,
So this is the
Zoomed out.
2.
Could you reach that one as well, or
which
not?
one, the white one?
Yes.
This one.
Possibly, yeah, that's where the starfish was.
Yeah.
Yeah,
Could you put in slurp 3 then?
yeah, sure.
You want to keep white.
We want them to keep separated to be sure we have two different individuals, because when they arrive in the box, they often break.
Fair enough.
Slurp.
Yeah.
Okay.
Okay.
And so it's up to HD. Get up. Little zoom. No, start to limit back over. We might be able to get it. I think we're a bit far away, are we?
See if we can.
No, I'm a bit far away back up.
Yeah.
Yeah, I might full extend there now.
Okay. Okay.
It's just a little bit out.
See if you can get it now.
Okay, inside. Take a mic check.
Even if we have a,
Yeah.
actually preferably if
Slurp.
we have a little piece, it's perfectly
There you
okay.
go.
Oh. Perfect.
That's it.
Perfect. It's
Okay.
in.
Yeah.
Yeah.
Okay.
Thank you, Patty.
Welcome. Zoom out on it.
I'm just taking notes and
So this was the slurp
3
method.
sponge.
It's in the holes.
And we are 1,
It's
2,
in the holes in the HD.
5,
Guys, look at the HD holes. Look at the holes.
Oh yes, it's in the hose.
Yeah, so suck it up again.
Yeah. Oh, there.
Well spotted.
Okay, that should be got through.
Good.
Okay. We have another system.
No,
So
no, we don't need
there's a system of boxes at the end of the robot with 6
that. 5.
5 compartments? You're right, 5 compartments, separated compartments. And when you slurp, when animals, they end up in one of these 5. And when the ROV will come up, we will open these compartments and retrieve the samples. And that's a really good way to get actually quite good quality samples, especially for amphipods.
And also to enable population genetics, so you know exactly where things were sampled from and not get all mixed with each other. We may continue to
Yeah,
try.
we are okay to continue the transect.
Okay. Bridge ROV, can resume the move, please.
I saw a little
Code
about
340,
gastropod.
continue
yeah,
at
I see
point
it.
1.
Thank you.
Sorry, I'm still copying notes. So
Yeah, I will write
3,
it.
4, 8.
So this species also, this little white bush, I like to call it the pom-pom sponge, will also be used for environmental DNA studies. So it's been discovered a few years back that sponges can be used as natural samplers for us because they're filter feeders. So they're constantly filtering the water, and doing so they actually accumulate DNA pieces that are in the water column. So they're actually accumulating DNA of every living organism in the area. So by extracting DNA from the sponge, you are actually extracting DNA from this whole habitat and environment. So one of us, Sergi Taboada at the museum in Madrid, is conducting eDNA studies on sponges, trying to see if we can get a better picture, a wider picture of the Schulz bank biodiversity, using sponges to help us doing that.
Shrimp.
Mm-hmm.
What's this white thing coming up?
On the rock,
Maybe-
you mean?
Yeah. Maybe an anemone. Next to tuna cat.
Yeah, there's another one here. It could be a sponge as well. It's becoming a bit more rocky here. You can see cobbles of volcanic rock, black rock. We are of course in a volcanic area, being very close to the Mid-Atlantic Ridge, and most of these seamounts are actually created by magma, which at some point came out from the Mid-Atlantic Ridge. Could you zoom on this?
Yeah, but we're fast. Maybe
We're going
we'll
fast.
make us
Could we
busy.
stop and just zoom on this sponge?
Bridge, are we all stop?
All stop.
You're fine?
No, I need a close-up. I'm just waiting for it to clear away a little bit. Let's see. It's sometimes very tricky, of course, to see things because of the sedimentation and the very, very fine clay sometimes that we find on these mountains. That is easily gets into the water. But let's give it a minute and see if this clears up. Mhm.
We'll stay in.
Yeah, well,
in the
maybe a
picture,
bit,
and we'll just zoom out a bit to get a better feeling.
or maybe a bit too close.
That's clearing up.
Yes. I have an idea of the species, but I'm not sure until I have seen the surface of that sponge.
What's flying in there?
Oh.
I'm not sure. It's just some
Mhm.
sediment.
Yeah, this looks like Geodia parva because you see all the warts on its surface. These are the oscules, so the openings for water running out of the sponge. And this warty surface is typical of that species, Geodia parva. This is a nice specimen. Geodia parva is one of our, I like to say, is one of our most social sponges because she loves to have other species sitting on top of her and overgrowing the surface, while other species of sponge really hate it, and they are smooth and naked, and they don't like to have other things growing on top of them. But this is Geodia parva, a very social sponge. What else can we see?
I'm sorry, I was not looking at the sponge, but it's a little crustacean
Yeah?
crawling back and forth to the left of it. You can just see the eyes.
Can you show me?
Yes.
Oh yeah, okay. Oh wow. Yeah, there's like a little hole on the left of the big sponge, and in that little hole there is a small amphipod. But of course you need a trained eye. I'm still not there yet.
It's a thing that moves and has pink eyes.
Yes, I see the antennas.
I mean, the most interesting thing in this entire image.
Of course.
But
Yeah. So let me log this amphipod and the social Geodia parva,
it's,
and we are at 1,051 meters depth. And we expect a lot more Geodia sponges as we will ascend this mountain. This is a very common species here. So we will not collect it. I have collected many before. Maybe on the summit we will collect one because I'm interested to see the other sponges growing on it. But here this one doesn't have a lot of things on it, so we will
okay.
leave it there.
Okay, we'll zoom
Thank
back
you.
out again,
Yes.
and then we will continue our journey.
But we are changing pilots.
Well, while they're doing a pilot change, I might as well zoom in again.
Mm.
You can see another purple star.
A lot of clams.
What are these branching little tree-like units?
This, I don't know. Could it be sponge spicules, like remains of dead sponges?
No, but these ones.
Oh, these ones, sponges.
The white, the white ones that look like trees.
Good to go?
Yeah.
We are good to go.
We are good to go.
Yes.
So this is getting more rocky. And sponges love rocks, hard bottom, to be able to have a point of attachment.
But also for their larvae to settle.
Exactly,
Many
yes.
of them distribute themselves via larvae, and they need any kind of hard substrate to settle down.
Yeah, yeah, and that's really. Thank you for saying that, because not so many people know that sponges have larvae that actually move in the water. So when sponges reproduce, quite often a larvae is produced, and that larvae can swim with small ciliae. It doesn't swim usually very far until it finds hard substrate, and then it can settle. There will be a sort of metamorphosis, just like the caterpillar transforming into a butterfly, but it would metamorphose itself into an adult sponge. So if you're joining us now, we are ascending the Schulz Bank Seamount on the western side of the mid-Atlantic ridge in the Arctic. And,
Asteroid.
oh yes,
And
a
then
nice sea star.
Ascidians. Shrimp. Hackle's just entering, otherwise you would have said something about this white sponge.
Oh. Oh. Probably a Roselidae.
With a feather star on top.
Mm-hmm. So these large white sponges are glass sponges, and I will not comment on them just now because they will become very, very abundant as we get to the summit of this mountain, and then you will see how beautiful they are. They will be everywhere. No, it's a kind of like overhang. Oh,
That's quite steep.
ooh,
Look at this.
wow.
You're okay until we hit
This is very special. Could we settle the ROV close to the wall so that we can zoom on the wall?
the yeah, you might get a bit of sediment coming with it, but
Okay,
could be okay. Yeah.
we're gonna try.
We'll give it a try.
Yeah. Let's see if there's too much sediment, we will have to leave. But this wall is very special, extremely rich. I can see a lot of little worm tubes, anemones,
Okay, bridge, slow to find one.
and sponges, of course,
Stop
and
here,
probably
Philip.
amphipods
Okay, get on.
in those cracks.
For sure, but the anemones look very picturesque. Like flowers.
Okay, zoom.
Yeah. Where do you want to zoom?
I don't know yet.
Sediment there now.
Mm,
Will clear, should clear.
okay.
We'll just wait a little bit.
This here from the T4 already alright, so we go there to here.
Hmm.
Lots of worms.
I think have we encountered so many worms before like this?
Nope.
That's the first time I see that type of community.
There's lots.
Yeah.
Clear.
Just park the properties.
So my colleague Sergi reminds me that these tubes are Serpulidae. They are polychaetes. So these are marine worms, polychaetes, and some of them live in little carbonate calcium tubes, and they belong to the family Serpulidae. This is what we are seeing here. It's very beautiful.
We should, if we could focus, well, a little bit further, we would see, like, at the end of each of the white tubes, there should be little filter fans coming out, and that's the worm feeding. You can maybe see it a little bit right in the middle here. At the lower part of the screen now.
Yeah, I can see them now. Yes.
Yeah, that's a worm. That's a worm.
Like a fluffy, moving with the current
Yes, they're also
tentacles. Yeah.
Yes, and when you touch them, or they're disturbed anyhow, they will pull back into their protection.
Yeah.
Into their
Yeah.
tubes,
The
as they
divers know them
go.
well.
All divers know them well, for sure.
Mm.
But it's interesting that there's so many tubes on top of each
Yeah.
other.
Yeah. This is very special. We are at 1,042 meters on the slope of Schulz Bank Seamount, and we've discovered this large community of tube Serpulidae, which is quite
And quite some bryozoans.
exceptional.
And
Bryozoans. These are the
Also-
little trees, white
and I'll
trees.
zoom out again.
And sponges, of course. These are the white specimens.
Now I'll move a little bit to our right side.
Yeah. I think there were really a couple of, there were really a lot of anemones.
Very good.
Oh, look at that.
I want to look.
Wow.
I want to go there
Yes.
and a bit further. What's
Mhm.
this?
That's probably a Roselidae
Was it
hanging from the
top? And
top. I think
ha-
it's growing weirdly.
And having a shrimp
Is
sitting
that-
on it.
Is that a sea star? Upside down, maybe.
And having one arm into the.
Yeah. Maybe it's eating something. So sea stars are predators. They're carnivorous animals.
But normally they eat with all arms attached,
Mhm.
and then the middle part goes up, or they turn their stomach inside out. Well,
Yeah,
and then
that's
they do
pretty
it.
scary.
Yeah, so it is like that.
No, yeah.
And they
So they bring
Their
their
stomach
stomach out of
out.
themselves and
Yeah.
place it on their prey
yeah
to digest
then
it
to digest it and
externally,
then, yeah,
and then they bring their stomach
suck-
back in.
suck it all back in and.
Yeah.
And normally when they do that, they attach their arms to the sediment or to whatever substrate,
Mhm.
and the middle part comes up. It's very unusual that you have one arm hanging
Yeah.
in the water. Is this a sponge as well that we're looking at? Right in the middle with the one
Yes, it is a tiny sponge.
opening? Very.
So again, the two lasers that we see very well here are 20 centimeters apart. To give you an idea of the sizes of these animals.
And almost all animals that we can see here, except for the sea star, are filter feeders or suspension feeders. So there are quite a lot of nutritions floating in the water
Yeah.
that they can filter out, or small other. Yeah, they mainly build crustaceans, but also eggs.
Yeah. I guess small fish could also be preyed
Yeah.
upon by the anemones.
Okay, I'll zoom out again, and let's go a
Copy
little
that.
bit.
Try that.
Go ahead.
And I'll just stay sunny for a while. It's quite nice view.
Yeah, okay, we'll do.
So we are now taking out our fisheye camera because this is a very nice place to actually have a very wide view of the habitat.
And I'm sorry that it's not possible to change screens, so we can't show you what this camera is seeing.
No.
We'll stick to this camera.
No. This is footage that we will have for the future to show the public, but we can't show it live. It's difficult to understand what type of substrate. It's like all the worms are growing on top of other worms, or is it rock? It must be rock in
Well,
the
it's a-
background, but it's difficult.
Like a porous lava.
Hmm? Should we try to sample this with a scoop or
No.
Won't be able to back
No.
over. We're actually
No.
too close to the wall.
or too close? Okay. Well, let's just take the position of this place because this will be an interesting place to come back to for scientists and study. Again, I've never seen a wall of tube
So
worms
this-
like this. This is pretty unique. And we are 73 degrees north between the Norwegian Sea and the Greenland Sea on the western side of the Mid-Atlantic Ridge. Here you have a view of the spotlights that are mounted on the ROV. Very powerful spotlights that enable us to give you this view, this wonderful view. You can see some rocks, definitely.
But you can also see a lot of empty tubes. Or at least
Yes,
not.
I agree.
I assume the white ones are
I mean,
where the active worms are.
Are tube reefs a thing?
Yes,
Could you have a reef made of tubes?
yes, you can.
Uh-huh.
And some cold water corals, that's actually a very similar structure.
Mm-hmm. Yes, growing on top of each other, on
Yeah.
the dead ones.
Yeah, and
Yes. Yes. And if you want to see an amazing deep sea coral garden / reef, you should look at one of our previous dives this week in the bamboo coral garden, which was a bit more south of here. It was an amazing dive as well.
let's continue this.
Let's continue. This is
This
very
is-
promising. Yeah. I'm very curious now to see how far it goes. Now you can see our fisheye camera here at the bottom. So we were at 1040. Okay, so the tubeworm wall is very localized, it seems. That was it. It was maybe 2 or 3 meters high, according to the depth I have in front of me. Very interesting. So now Ascidians are getting
Ascidians.
very common.
Dominating this.
Ascidians are like the transparent tubes you are starting to see on your screen. These tubes have 2 holes, one for incoming water, one for outcoming water. They are also filter feeders, just like sponges. They filter the water and feed from small, very small organisms or organic matter that's in the water column.
Well, I think this is a more,
Oh no, but
Oh.
wait, it's starting again.
yeah. And I think
Oh.
all of these little white ones are the polychaetes.
Hmm.
Yes. Hmm?
So there seems to be quite a lot of sedimentation on this part of the mountain. Hello.
Hello.
Hello,
Hello.
Jenny.
Do you want to go to eat or shall I
Whatever
go?
you prefer.
I can go and then I will come back
That's
later to, and then I'll probably sit because I want to see actually
Yeah.
what
yeah,
happens
we can-
next.
Just
We can
come
be.
soon, then I'll go when you're back.
Yeah, yeah, sure.
Superb.
We do that.
Thank you.
You're in good hands now with Jenny.
Thank you.
Thank you. Hello, hello. This is beautiful.
It is.
Oh, is that a crinoid up here? It's not a pycnon.
Okay, Bridge, can
No.
you resume that move, please?
It's too steep. I
Penny
can't stop.
340,
Yeah,
continue
yeah.
to point 1.
Thank
That's
you.
all good.
And we've just encountered a wall full of polychaetes.
Okay.
The entire satellite city.
Tablet City. So
still quite steep.
Grenadier. We can have a look at the profile if we've got it. Yeah. Lots of Ascidians. So we are currently diving at 1028 meters depth at Schulz Bank, where we have already completed one dive and are now with the second, right in the middle of the second dive. And we see a lot of soft
Would it be
bottom.
possible to sit down here and use a coffee mug, or are they too steep?
Too steep.
Okay.
Okay. So we're facing quite a steep slope here. Sometimes it's hard to see in that view of this camera. But it's too steep to sit down for the ROV.
I'm talking about a coffee mug to the pilots, and it's actually a little gear unit, which of course looks like a coffee mug and has a net attached. But we might be able to use that later. And there's a tulip sponge.
Yeah. Oh, I wrote lollipop. Maybe it's
Lullaby, you're right. Maybe it is.
because the tulips were more
You're
like.
right. You're right. And so many
Oh.
birdle stars. I'll just zoom in again, and you see all this
And this
pink
one
dots on the floor. You can just see them. They're hundreds.
Oh yes, wow. I logged Bicydendurix and so ophiurids are also called brittle stars, and they are sister to sea stars and sea urchins and feather stars and sea cucumbers. They are all belonging to the same group, which are the echinoderms.
Yeah. What unifies all this group is the so-called pentamere. You might know it from the Pentagon, having a structure of 5 angles, which is for sea star. It's very obvious, but for an urchin or a sea cucumber, you have to look quite close.
Turning that star. Yeah. Yeah. See.
Can we stop here?
Yeah.
Or slow down, just so we can use the.
Yeah. Let's slow down first. So.
Thank you.
Wanna zoom in?
So you can actually see the purple stars, and you see all the tubes.
Oh, look at it move. So we see tubes, we see brittle stars. Do you think the white tubes are sublet tubes and the brown tubes are these agglutinated foraminifera?
Yes,
Mm.
but also on the wall, we just had brown tubes, which were also from this worms, but not with a living worm anymore. So on the wall we had both, but these are the foraminifera.
Yeah. So there's an ophiurid or brittle star just to the lower left corner.
Yeah. And to the left, right, and in the upper right, there are two.
Yeah, they're buried.
Yeah, I'll zoom out again.
We can go to that one.
We can just fly on.
We
yeah.
can fly on. We can
Yes,
fly
let's
on,
fly on.
thank you.
Thank you.
So we're climbing the slope because we want to go to the top of this seamount. So we will have a gentle incline for a climb. That's what I want to say. A gentle climb for maybe another 100 meters, maybe a bit more. And then we have some sponges. Do we know what they are? And we have huge crinoids, which is nice. The ventilation is really strong today, isn't it?
It's quite chilly here.
It's we have a ventilation system in the shack because there's lots of PCs and whatever, like lots of things that need to be cooled down. And it's quite strong actually, which is why at least I am always dressed really warm here. Are these small asconemas? I think so.
What's an asconema?
An asconema is a sponge, a glass sponge that I would say is a vase shaped, and we just saw it on the seabed. It's a species I learned during this cruise because we have sponge taxonomists on board, and I didn't know. I barely knew any sponge species names before this cruise, and the knowledge is increasing. I think this would be another asconema with a crinoid. Really beautiful.
Crinoid is a feather star.
Yes. Asconema.
Another lily.
Assyrian?
I wasn't sure.
Oh, can we zoom in on that? Oh, it's okay. Just, I was interested in the yellow, but this is nice. This is very nice. Anemones. Obsidian. So
I'll zoom out again.
the seabed is getting more and more rocky. It's quite a steep incline here.
Can we stop in front of the rocks coming up?
Yeah.
Oh.
Because I would like to look out.
Oh my God! What
That's
is?
exactly? That's why I would like to stop.
Okay, this is so beautiful. We have an incredible overhang here, which I will log exactly as I just said.
Same area.
Wow, we are at 1,004 meters. Wow, this is full of life because the current just hits it. Look at this soft coral. Oh my God.
So we have soft coral, we have an overhang. It's a very big rock.
Bridge RV.
RV bridge.
Stop. Can we stop for a moment, please?
And see all the lollipop sponges on top?
This is so beautiful.
Very pretty. As soon as
Amazing. What are these? Look, can we please go?
yeah, just a second. Looking here first.
So we have carnivore sponges. Lots of subbalids.
On the outer parts of the screen now, you can see all these carnivore sponges.
Yes.
The pink ones are the anemones. You can see an anemone also sitting on a sponge. There they are attached to a rock with several ascidians. And all these white tubes are our polychaetes again, our cerbalids.
Or bristleworms.
Yes, bristleworms. And they're having their tentacles in the water. Did you see the sea star?
Yes. I have looked it.
Ascidian again.
And this is so rich. And I think it's so interesting because we just climbed the slope and it wasn't poor in life, but as soon as you have a rocky outcrop with a bit of current, more current, it just gets so diverse and all the species that need hard bottom just settle. It's incredible. So here we have a huge feather star. The lasers are 20 centimeters apart. Are we
Quite-
moving?
Yeah.
Well,
Oh.
it's quite a current.
Moving back in.
Yeah, okay.
We'll zoom out again.
Yeah, but
Well, it's quite hard to stop it. Look at this soft coral right in front of us.
can you hold her steady here? Is that possible?
That's
Yeah, we're
okay,
still
yeah.
moving.
Thank you.
Thank you. We will zoom in again. You see, I'm impressed by all these lollipops.
There, it's Lollipop Garden.
And here we go.
And this soft coral, wow. What is there?
So I'm trying. I do not know.
What?
What is this red blob just in the middle?
Is that a sea cucumber?
No, I don't think so, but I don't know.
This is Asterias. What? And we have Aspestopluma. Is it an ascidian?
And you can also see the anemones. The not pink ones.
Yes.
The white ones. So you have two dominant species here.
This is beautiful. Can we turn off the lasers for a second, please?
Yes.
Thank you. Wow. Amazing. So sometimes we have the lasers on and sometimes we have the lasers off. And we do that. So usually you always have the lasers on, so you always know you have always a size reference, you always have a scale in your image. But if you really want to capture beauty shots of the habitat, of the fauna, of the diversity that we are currently seeing, it is nice to not have the laser points in the image, which is why we first film it with lasers on and then we take them off, because we have the scale on another image that can be used for science, for analysis.
What we saw at the top was an animal. Let's see if we see one from the side.
You're right, you're right.
And it's a dark red color.
Do you think we can get a zoom? Or maybe let's just stay like this. That's a clam, isn't it? Wow.
I'm not coincidentally centering the amphipod, by the way.
Yeah, obviously.
So
our amphipod specialist, Annalena, has, of course, her eyes just center on the amphipods right away that you might not even spot that quickly as she can do. I mean, you're so quick at spotting them, it's insane. They are everywhere and I know it, but you always find them straight away. It's incredible. I love this.
yeah,
It's so
Gorgeous.
beautiful. Wow.
it's quite interesting that these filter feeders are on both sides of the rock and hanging down and having the lollipops on top
Yeah. So
of the structure.
filter feeders.
Would it be possible to dim the light? Please. I have to see if there's something glowing going on.
The deep sea is full of life, isn't it? It's so cool to see this.
Thank you. Okay. You could turn them back on. Thank you.
Yeah.
Do you see the shrimp on top?
No. Yes, now I do.
Maybe close your iris.
Yeah.
Close the iris a little bit.
I prefer not to do it while the sexton is out,
No, okay.
so they will.
No, that's fine.
I really want to zoom in there.
We'll get to the next one.
That's it.
There.
Shall we go a bit further down?
Is it possible to go down with the rock a little bit?
Just like
A little bit?
teeny tiny bit.
Thank you.
There it's really rich in anemones and soft corals. It's incredible. There's a colors on days on the seafloor and we
Yes.
want to zoom in on that one.
Yes, I would actually like to.
Oh my god.
There's another one next
There's
to
a
it.
huge,
There are three actually.
there are
There's-
three
there's
huge
three, and
sea
the-
spiders.
Sorry, we have to move our eyes away from the rock to the floor.
Yes.
It's just now in front.
Oh
Can
my
you please pull the sextant camera back in?
Please.
It's just, just behind it is this big sea spider. Thank you.
Amazing.
And we wish to focus on it, just to have a zoom. You see the sea spider here with its color legs?
Yeah.
And this is what I
There's
would like.
another one.
Yeah, well, there are three in the picture now.
Which one?
No,
No,
this is a
no,
print.
yeah,
This is
but,
a
but
print.
we will focus on this one. Sorry, I'm getting it centered.
That.
Can we turn the lasers on just for a second for size, please? Thank you. You can go off again. now.
So this, we first thought we saw 3. Actually, it's 1 because the other is a crinoid.
This is a crinoid. You're right. It is not a.
So what we are seeing here is a sea spider, and the species name is Colosondes proboscidea. It's a very large sea spider. Have we measured it with the lasers? So
Yes.
it's more than 20 centimeters in diameter, and it is just an incredible species.
Oh, look what it's doing.
And it's called proboscidea because it has a very large, very pronounced proboscis, which is
That's the yellow part, and that. I'm losing it.
which is the yellow. We're losing it a bit now, Rob.
Sorry?
Now it's out of the picture. There. Now we're
There.
back.
We're back again.
Do you see
Thank
it?
you. Yes. It's this one.
Thank you.
I have the rock just beside me, so it's not-
Yeah,
Yes.
yeah, yeah.
Yes. No,
yeah.
we appreciate
that's
you.
all, yeah.
Thank you very much.
This is already. We're having this big rock ahead of us or above. We're talking about the proboscis. That's the yellow part. And you've got the central unit.
And there's quite some current here too. So
Yes. But you can see how colorful it is.
Yeah, amazing.
On
I've been
quite
watching
prospect.
this from the dry lab.
So yeah, you happy?
it's really
I'll zoom
cool.
out. I'll zoom out again because this is a bit tricky. We're under this, just at the edge of the rock.
Mm-hmm.
We would like to stay here and then have a look at the rock.
That's what we actually came here for.
Yeah.
but we
Oh,
were-
wow.
Yeah,
Yes.
exactly. We were
Oh
just
my
distracted
God.
by this. Look at this.
Oh.
It's covered with carnivore
rockling.
sponges. Oh,
Yes.
I can
We noted.
Yes.
We noted those.
I can talk
And,
about that a little bit. Yes.
Well, you can also talk about this lollipops, which are. This is a big rock, and that you can't see them now, but on the top are the lollipops.
Mm-hmm.
If we can just stay at this.
Rob is trying, but our
This is
place,
amazing.
we would just like to
to stay
hoover
like
here.
to
just
stay
for-
like this is amazing.
okay.
Yes, thank you.
Thank you.
Thank you.
Very lovely.
It's like an upside down garden, you know?
Yeah.
Ah. It's incredibly beautiful. Oi! What's that? What is it? We need to zoom in. I'm sorry.
Sorry, we have to go back to the sea floor.
We just need to zoom very quickly.
Yes, we will. Well,
Night.
I can't.
Night.
Yeah, I'm trying.
What is it? What is it? It's a feeding zolaster. I think it's a zolaster, and I think it's actually feeding on an anemone. So it was a feeding sea star, and I think
Just gonna sit down.
of the genus
Are you going to
zolaster.
sit down?
Yeah.
Oh, that's much appreciated. That's really brilliant.
I don't want to rush you, but you have 15 minutes to
Okay, I'll be back.
get dinner.
I'll be back soon. I left you a great spot here.
Yes.
Okay,
Thank you.
now we are sitting.
Is that any good? If not,
Yes, thank you, Rob.
it's all right.
I mean, if we could,
Slew the headroom.
but if it steers up a lot, then maybe we just sit tight. It's a bit like
Okay, just try that.
Have you collected anything
no,
in the meantime?
no.
Okay.
We discovered the rock and that was it. You know us. It.
Okay. Yeah. No. I get it. It's beautiful.
Yeah.
Yeah, cool. Do a look there, so
Let's just stay for a bit longer and admire this highly diverse rock. I love this.
It's so
I
strange
love
to have
this.
this boulder in the middle of the slope.
We can go closer. Can you move center the camera a bit, please?
Sure.
Thank you.
How's that? Is that good?
Yes. Is it possible to move a
Maybe
bit
To be a
closer?
bit closer, yeah.
Because we would really love some zooms.
Oh, you want to look at the rock?
Yes, please.
Yes.
I thought that was... Never mind.
Maybe we can sit here to the. Yeah, you'll find a good spot to sit her down. I really want to zoom here in this huge anemone. Oh my god. We just have to stay here for a bit
Yeah.
longer. Look how it's clinging to it.
Yeah.
The rock. The fish. It's really strong. Thank you so much. Okay, we just need to have a beauty.
Let's just enjoy
Oh my
this.
god, let's just enjoy this beautiful garden of Paco. What do we see?
Lots of anemones, different species. Of course, sponges that are a bit more. Well, there's a big white sponge. We think it's Helicondria.
Helicondria. You know what? Helicondria is one of the sponges that a nudibranch that I worked on really likes.
Yeah,
Oh,
they're good food for mollusks. And then all the feather-looking animals that you see are sponges too, but they're carnivorous sponges. So here you have the genus Abyssocladia and Cladorhiza. And they catch prey. These are sponges that do not filter feed like other sponges. Instead, they actually catch small crustaceans, small shrimps. They have in their skeleton, they have little pieces that are hook-shaped. And when a small crustacean, a small shrimp, for instance, will swim close to it, it will get entangled in those little hooks. Some will get away, but some will get trapped. And then the sponge cells will migrate towards the prey, surround it, digest it externally by releasing lots of enzymes, and it will be eaten on the surface of the sponge, and then the leftovers will be just remain, and then the cells will go back to their place. And that's how carnivorous sponges eat. And it's so crazy to find so many of them here, with so many anemones. It's really unique.
you look at this.
Oh, and lollipop sponges.
yes.
on top.
We can zoom in on them.
Yeah, absolutely.
Go ahead.
So that's the genus Stylochordilla, and it's the species Stylochordilla borealis. Look at those. Look at this. This is a really cool sponge
Yes.
that keeps its babies inside. So it's a sponge that does direct development, meaning that the babies grow inside the mother and then are released as little tiny lollipop sponges. So they must be super cute. I've never seen them actually coming out of the mother, but
Aw.
little lollipop like this must be super cute.
Yes.
It's pretty rare in sponges that you have this direct development. Usually, I think I mentioned it earlier, the larvae swims in the water and the larvae settles elsewhere. But here it's very different. It's actually little babies coming out from the mother sponge.
Look at the current. Look how strong
Yeah.
the current is.
Yeah.
Do you think that is a sponge? The animal that is wobbling?
I think it is. I think it's probably Cladorhiza, but
It's
yeah,
a bud?
maybe it's a smaller individual.
Ah.
Oh, we have Ascidians. We still have the tubes, by the way.
Yes, sublids.
They're still here. The tubeworms, Serpulidae.
This is so rich.
Yeah.
And I bet if we would go even closer, we would find, again, huge biodiversity. Like the tiny critters, they're everywhere. I can promise you that. nudibranchs in this current for sure,
Mm.
but we can't see them because it's even more tiny.
Sorry,
No worries.
No worries.
slipping.
Okay.
Yeah, I could feel the image was moving and I wasn't touching the zoom.
Yeah.
So it wasn't me. I felt it was the ROV.
Are we still good, guys? To sit a bit longer?
Okay.
This is so, it's like a reef.
It's amazing.
It's
It
like a
is-
sponge garden.
It's incredible. I've called it anemone heaven
Okay.
in the log, but
understand.
sponge garden,
No, I
anemone
understand.
heaven, anything works.
Yeah, it's a
like
lot of it,
really.
yeah.
Look at the soft coral on top there. Isn't
Oh,
it
yeah.
incredible?
Yeah, to the top left.
Let's see if we can get the entire rock into the picture for a second there. Wow.
Let me just
Yeah, go ahead.
there. Like this, we have a
Exactly.
full view. So, the deep sea is colorful.
Oh my god.
We often think the deep sea is dark and gray, and no, it's not.
I could stay here forever. You know, when I'm diving,
Mm-hmm.
if I would encounter this during a dive in shallower waters, which is like with different species, but when you see rocks like this, I just hang there the entire dive. I don't move anymore. I'm
Mm.
done. I have everything I need. Seriously.
Mm.
Wow. Let's do one last zoom.
Yeah.
You choose.
Mm. Well, I'm gonna choose those carnivorous sponges. Oops, that was a bit fast, but
Yeah.
I'm eager to look at them.
Yeah.
Look at, oh, there's more lollipop.
Yes.
It's like a little tropical forest sponge.
Look how the feather star is clinging onto,
Oh, yeah,
I don't know, a hydroid maybe, or
Yeah.
a coral.
I think
or-
they're actually still carnivorous sponges, but skeletons
Ah,
maybe that are
ah.
dead. Yeah, I think these are remnants.
But it's really hanging in there. It's
Yeah.
like, oh, the current is strong.
And, oh, that was an amphipod,
Yeah.
right?
Yes, you're right. You're good.
Yeah.
Well spotted.
I've been hanging out with you and
Yeah.
Annalena for now a while.
Amazing.
Wow. Yes, and I think it's a good thing you mentioned the currents, because again, people think the deep sea
Mm.
is like a motionless desert, nothing happening, and
Right.
this couldn't be the opposite. Like we have currents, we have sometimes lots of currents actually along the year. It changes.
Mm.
There's different seasons when there's more food coming from the surface or less food. Temperatures are changing.
I was just about to check the temperature.
Mhm.
We have a minus 0.2 degrees Celsius.
There you go. Yeah. So that's pretty much the temperature we've had so far. Even colder.
Even colder.
We've had colder at 2,000 meters, minus 0.6, I think.
Right.
So we're in
Okay, move.
minus temperatures. Oh, we're moving. Yes.
Yeah,
Okay.
and it's fine. It's
Yeah.
fine. We're done here.
Yeah, just slip right off.
That's all right.
There-
Just
Good to
Yeah.
zoom out.
I'm going to zoom out.
Yes.
There you
Oops,
go.
All good. Look, it's still there. Kolla Sundays, Proboski there.
Yeah.
Okay, we're flying off.
We're ready to move on.
Yes.
Thank you.
Yep.
Thank you, Rob.
The currents are really strong, so it is. Sometimes it's not easy, and also with this overhang, the pilots have to make sure the ROV is not being dragged under.
Hmm?
So it is very challenging to fly here.
Oh.
So we had a very quick lift off now because it's easier than to just move
wow.
away.
It's just a huge slab of
It's amazing.
volcanic rock.
Amazing.
Maybe it comes from a downslide or
Exactly.
a landslide, sorry.
Yeah.
No, very strange.
If you can turn the lasers back on, please.
Yeah.
Cheers.
So I'm back in the shack.
You are back in the shack?
That was spectacular.
Yes.
We're still on Schulz Bank Seamount. If you're coming, viewing this now. It's an Arctic seamount on the western side of the mid-Atlantic ridge. Yesterday we were at Loki's Castle, an amazing hydrothermal vent site discovered in July 2008 by a Norwegian expedition. And while they were doing that, they explored one of the nearest and most impressive seamounts, which was the Schulz bank. And that's where we are today. This is a huge mountain, about 2,000 meters high, and that we are today ascending. We started we were at minus 200 meters, and now we're at 1,000 meters. So. And if you want to see actually our live dive from yesterday, which was amazing,
Oh my god, really.
especially the end, we won't tell you what's at the end of that dive, but let it see for yourself.
You gotta stay until the end.
Yeah. Yeah, you can definitely see that, rewatch this on YouTube, so.
Yeah. So what I've logged now is basically we're back to sedimented slope with some
Yeah.
ophiurids, anemones, ascidians, some lollipops.
Can we start moving the ship bearing 330 0.1 continuously, please?
330.1 continuous.
Possible? Or it's too steep to collect one of these two?
Collect one of them?
Okay. Sorry, Reykjavik.
Bearing 330 at 0.1 continuous.
Don't start the movement. Sorry.
Any by.
Thank you.
That's interesting.
Mm-hmm.
What do you want to do? Suck it or
Suck it is fine.
slurp it?
Yeah.
Let's see if
And that
that's...
would be slurped one would work.
What is in there?
It's
That might be...
Lisodendroids.
what do you think? You can have a look at... No, go for it.
So
No,
we're
you-
sitting down. We were about to fly off, and then
Sorry, I saw more lollipops sponges, and they're much more accessible than they were on the amazing rock previously.
Okay, ready when you are.
And I'm going to explain a little bit why I need a specimen. I just told you that they do direct development,
Please.
and that this is very rare in
What
sponges.
What tank? One.
Yeah, slurp one.
are we
Thank you.
sitting?
Okay.
Are we sitting? So okay.
So I will
Um,
start
See the arm.
to make it, maybe zoom a little bit on these two lollipop sponges.
Let us just have a little zoom and
Yeah.
chat, and then
Yeah. Oops.
it's more
too much.
directed towards Brock, actually. But no worries. We will just have a little zoom in and a little chat about them, and then we can collect one
It's fine.
one specimen.
Very cool.
So they're very poorly studied, especially in the Arctic, and because they have this really unique way of reproducing themselves, we have a project where we try to understand how the larvae develops inside the mother. Do they pick up bacteria from the mother? And basically the development of these larvae. And for that, I need samples that I will use to do histological sections to look at the anatomy of these little babies developing, to try to understand. Understanding the reproduction of sponges is essential to preserve them, because then we will know at what time they reproduce. Is it all year long? Is it only during the spring? And knowing how they develop also, because then we will know how many babies they can make. We can also understand exactly what they need for reproduction, how much food, how long the babies can live, and we can actually know that by how much, by measuring how much reserves of energy is stored in those little babies. So many questions we can answer by subsampling one specimen. I have subsampled another specimen on another seamount previously, on the previous dive, but I haven't sampled any on the Schulz bank yet. So we are going to try to do this with the slurp, the small vacuum cleaner type tube, to try to have a really clean sampling.
You know what? We sit at exactly.
Oh, we
now
are.
it moved, but it was 1,000.0
Yeah.
meters.
Yeah, we are precisely at 1,000 meters depth under the surface. So the water's minus 0.2 and the pressure is 100 times the current pressure you have while you're watching this. So people living like us at the surface, there is 1 bar of pressure, but when you go deep, this increases 1 bar more every 10 meters. And this pressure that's coming is from all the water that's above you. And because we are at 1,000 meters, the pressure is here 100 bar, which is huge pressure.
So pressure's on. Let's go.
Yeah, let's go for collecting.
Is it
The closest or easiest for you? Yeah, let's take this one.
steno? What is it called again?
Stylocordilla with a Y. Yeah, exactly. And then with another Y. D-Y-L-A. Yeah, exactly.
That's. And it was the slurp one.
Yeah, then I'll put it here in yes, one.
Okay.
So it's cast 6.
What was cast 2? Because it.
Oh, cast 2 is actually this complicated.
Because here it says cast 2 extreme.
Nope, that's a mistake. That should be slurp 1.
Yeah.
Sorry.
It's so tiny.
Oh my god, it's attached.
Aye.
Mm-hmm.
Aye.
Oh, no, still there.
Yeah, it's still.
Yeah.
Wait, and stop.
Stop.
Yeah.
I think we have it.
Yes.
Yes.
We do. That was really.
Interesting. So it must build roots.
Yeah, it was really.
It must have little roots, and that is an interesting fact that I didn't know.
I will make a note of this.
Yeah. And I mean, it makes sense. We just said there's a lot of current. They need
Yeah.
to be somehow attached to the ground firmly. And I'm actually happy we got a bit of sediment, because then we will have pieces of these little interesting tubes at the surface that I've been curious about as well. So we will use this material also to study that.
Do we want any other samples or?
No, we're good. We can continue.
Actually,
Thank you very much. Oh,
just one sec, because I
Yeah.
saw a very interesting snail in the background that I just quickly want to check out.
by all means.
Just need a little mollusk content there before we head off. Yeah.
Ooh, look at that.
Hello.
Oh. Do you still want to see the snail? No.
No, now the sea
Now we have
star
everything.
this is very pretty.
Yeah.
There's lots going on.
Yeah, sea stars love to eat sponges, so I don't know if this one is going towards that lollipop sponge or another sponge, but Of course, we won't wait to see what's going to happen, but it's nice to have this little scenery here.
It could be a Tilasta, but I'm not sure. It could be another species. I don't really know. This is back to you. Okay, but I think we can continue.
Yeah, okay. we're good to go.
You start moving.
Oh, look, there's really lollipop sponges a bit everywhere.
Yeah, amazing, really.
Wow. But you know what? It reminds me when we had the drop-down camera dive, and we started to see these lollipop, and they were in a similar habitat, very sandy and so on. So they like those sandy plains or slopes.
Really? Yeah.
I guess. I mean, I don't know. I've
Can you center it?
sure.
Thank you.
Yeah.
Thank you.
Yeah.
Yeah, it's really, they really do like these soft sediments. There's a tape change in 5 minutes coming up.
Okay.
We are, of course, recording all of this because these movies, this footage will be studied by, analyzed by biologists.
Bridge RV.
RV bridge.
Can we have movement of the ship 330 at 0.1 continuously, please?
Okay, 330 continuous at 1.
Thank you.
What is that?
That's another Hymenasta.
Uh-huh.
Yes, they don't necessarily are. Sometimes they're really red, but here they are more whitish.
Mm-hmm. Oh, could they be babies of those?
Yes. Oh, they're so.
And this is the one we
pretty.
collected,
yes,
right?
Yes, we
Yeah.
collected one specimen, and it's of the genus Hymenasta. It's a sea star, and they are very beautiful. They have this, as you can see, their arms are interconnected by a skin-like structure.
Mm.
Oh my God! There are so many highlights here today. We just.
Yeah, but it's pretty much the first time we are on a mountain slope like this. Look at this.
Let's zoom in.
Can we stop and enjoy these
Sorry.
lollipops?
Yep.
I'm just gonna zoom on them once you're
Bridge RV.
stabilized.
RV bridge.
Can we stop the movement for now, please?
Is it an
All stop.
It's
Anemones.
covered with
Yeah.
anemones.
So it is known that the stocks of sponges are a very demanded habitat because as soon as you're on a stalk, you can lift yourself from the bottom of the ocean and therefore have more currents and therefore have more food.
Yeah.
But the previous lollipops we saw were kind of naked in that sense, but no, these have a lot of friends. Beautiful.
Should we turn off the lasers for a second for a
Yeah.
beauty shot?
Can we turn off the lasers?
Yes, sir.
Thank you.
This is incredible.
And this one has a brittle star, or
Exactly.
Yeah.
So we have at least one species of anemone that
Mhm.
are really clinging to this very thin branch, or what would stalk, to the stalk of the
Mhm.
lollipop sponge. And
we now know that this stalk is very
yeah,
strong.
yes, yeah, really.
Let's see if I can zoom in a little bit more, actually.
We can. Wow. Oh, there's a tiny whelk
Where do you see it?
to the right.
Oh, wow,
corner
yes.
Up there.
So that's a tiny shell. Yeah, you're right, on the right
Yeah.
upper side of the screen. But I was totally focused on the anemones.
Yes, obviously.
It doesn't harm the sponge at all. They're just associates. They're both feeding in the water column, not necessarily competing for food. Sponges eat really tiny particles, bacteria, and particulate organic matter. Anemones, I think, are looking for more juicy bits in the water column. So it's a nice association. Maybe they protect them from sea stars, or maybe sea stars eat both. We do not know.
I think sea stars eat almost anything they encounter.
You know, I always get asked what are the apex predators in those habitats, and I think we've discussed about this.
Yes, we have.
So I usually say sea stars, but I don't know. Apart from fish,
Yeah, you know, if Anna would be sitting here, her answer would be amphipods for sure.
yeah, that's true.
Are we good?
We are good.
Do you want to change the tape?
let's-
Yes, please.
Yes, let's change the.
Let's just do it now.
Okay.
Yeah.
Thank you for reminding us.
1, 2, 3.
Mhm.
Okay, so we're just
Thank you.
thanks.
When you're ready.
Could you tell in
Yeah.
the dry lab
Ready.
that we've just changed it? We take.
Probably heard you.
Take
Yeah, but
1,
sometimes,
2, 3.
They're dry lab, are we?
Recording.
Right, lad?
Yeah, we've just changed the media disc.
Yeah, do the same.
Okay.
All right.
Thank you.
We may continue the move with lasers on, please.
Laser on.
Thank you.
That bridge, RV.
RV bridge.
Yeah, we can continue that move now, please. 330.1 continuous.
Maybe we managed to actually
Okay,
continue.
330, continuous at 5. Okay.
Thank you.
There's always something interesting.
Yeah.
And but this was really worth it because
Yeah.
this close association between the stems of the lollipop sponge and these anemones really clinging to it. It's there again.
Yeah.
So that was really nice to capture. And interesting, not only interesting, but also biologically important to capture these species interactions.
Yeah. You can only do that by going there, in their habitats. So we feel very lucky to have the opportunity to do that. And that's also why we want to share it with you, because, I mean, this is rather unique. So these lollipop sponges are also interesting because they have a counter species in the Antarctic that looks almost similar.
Yeah.
And it's called the Chupa Chups sponge. This name was given by the discoverers and describers. It was discovered and described not so long ago, actually, in the years 2000. So it's the lollipop sponge in the north and the Chupa Chups in the south.
That is really funny.
So as you can see in the Arctic, there's not that many corals. We don't really know why. Maybe it's too cold. This is not an environment that they where they can thrive. There are some corals though, because we've seen them on the previous dive on Bamboo Coral Reef, right?
Yes.
That was 2 or 3 days ago. I'm now losing.
I have lost
track of time.
Yeah, me too. Me too.
But if you want to see that dive,
yeah.
please go and see the Bamboo Coral Garden dive, which was amazing. And then you'll see corals.
Well, there were a couple of soft corals now on this beautiful rock.
Ah, yes, that's true. Yes.
Quite big ones.
Yes.
But
Not hard corals, though.
no.
It's true that when I say corals
Yeah.
in my mind, I'm thinking hard corals,
Right.
like the ones you would see in coral reefs. But yeah, it's usually sponges that dominate in these habitats.
The view we're currently having on our camera is the view we would stick to if this would be a strict transect, where we wouldn't zoom or stop or change the camera setting, but just go in a straight line to monitor the seafloor and its biodiversity. We would just stick to about exactly this setting. So you always have the lasers visible and you keep a very steady speed of both the ship and the ROV.
You need the scale
exactly.
and the angle of the camera
Yeah.
to be the same.
So this is a very scientific setup we're currently
Mhm.
having. But because we want to show you the beauty of the seafloor and of the deep sea and zoom in and explore, we always have sections where we do that, and then we have sections where we keep it a bit more steady. But usually you would go like this for several hundred meters, right?
Mhm.
Without disturbing the camera, basically. So there are different kinds of setups how you can design and perform these ROV dives. It just occurred to me that we
Yeah.
have this,
Absolutely.
yeah.
So now we have a terrain with lots of little pebbles, still volcanic rock. And ooh, we start to see some golf ball sponges. The orange ones, the little orange little balls that are appearing on your screen. Ooh, there was a shadow.
Yep, there you go.
Oh, there.
What? That
Okay,
was-
sorry, we have different cameras, and so we just saw a big fish that was flying over the ROV, but you all only saw the shadow, but I saw it on the different screen. It was a fairly large fish.
Yeah, it was dropping by like very briefly. It's just a slope ahead of us, right?
Yeah.
We can leave it.
Yes, so you will start to see those little orange balls. I still see them. They are the golf ball sponges. And they also actually reproduce with direct development, just like the lollipop sponges.
There's a moving whelk, I think.
Oh wow, yes.
We will not stop for that. I'm just excited. But that is so cool. Paco showed me in the lab. We had sampled a golf ball sponge.
Mhm.
They're called craniella,
Yeah, exactly.
and when you can slice them in half. Maybe you want to tell this.
No, it's good that you tell it.
He showed me for the first time, and I had never seen a sponge been cut in half, and it's so beautiful because it has this, how do you say, it's not a cuticle, is it?
No, and it's not, no organs,
No.
no tissue. It's just a mass of cells, actually, but they are organized with a skeleton, a glass skeleton, because sponges are the only animals that have a skeleton made of glass.
Yeah,
Yeah.
it was just really beautiful.
And then I showed you the little babies that were inside.
Yeah.
Yeah.
So you can really see it, and it's, yeah.
So we're trying to understand actually when the golf ball sponges reproduce. So I've been collecting some at different times of year. And it's the first time I collect in May. So this is a fantastic opportunity. And they reproduce still in May.
Yeah.
So actually a lot of sponges start reproducing in May. Yeah.
Look at the whelk. Ooh, and there's a feasting. Can we hold here for a sec? Is that possible?
Yeah.
Thank you.
Is it very steep here? Or it's okay?
It's reasonable.
Okay.
What do you want to do?
No,
I
just
just
to
want
know-
to focus on 2 animals.
Yeah, we want to just zoom.
I think you've got these two here.
So there was snail that was a bit lower.
We don't need to sit. If you can hold her steady, it's fine. If it's
Oh, you really have good eyes for these. I still no, my eye is not trained for
yeah.
the gastropods, small snails.
Bridge RV, can we stop the movement for now, please?
Sorry for the movement here. We're just settling in. So we have 2 things that
Oh, yes.
that caught my eye.
Beautiful.
That's good.
First of all, it's this buxonid snail running away from us, you know.
That's what
They can really be fast, actually.
Actually, these are predators as well.
They are.
Yeah.
They have a very strong rasping tongue, and they would just go on and, you know,
So
rasp like anything they can find from rocks, and they eat small invertebrates that live in the sediment. And they can sniff with this. You see this little tube
Yep.
sticking up?
Yeah.
That's called siphon, and they can really sense their environment well and sniff up prey. So once we deployed traps to collect amphipods, actually, with some very smelly bait, and all we got were these snails that were just rushing towards it and feasting on the bait that was actually meant for some amphipods. What are we looking at?
An anemone?
It's an retracted anemone. Okay, all good, all good. I was confused. Okay. Anyway. Okay.
No, but it was nice to
See the little.
make a stop for the little snail.
Sorry, I'm confused today. My left and right is off. Now we are back in the middle. Yes.
We can continue. Thank you.
Bridge RV.
RV bridge,
Can we resume movement?
RV bridge,
Can
Oh.
we resume movement, please?
go.
Thank you.
This is nice because zooming out, we can really see the vastness of
Yeah.
this slope.
Yeah.
Really nice.
You start to see those potato ascidians. They look brown. They have 2 holes, and they're at the bottom, attached to the bottom as well. So there's always that little yellow sponge showing off.
Do you know what it is?
So we've collected 1 specimen earlier. We're not sure, but it's probably one of those sponges that we call keratose sponge. They don't have a skeleton actually made of glass. Some sponges do not have a skeleton made of glass, but they have instead a skeleton made of spongin. Spongin is a type of collagen that sponges have invented. They're the only ones to have it, that type of collagen. It's called spongin. And when you use a bath sponge, this is what gives the bath sponge its...
Look, it's a tumbling.
Oh, yeah. Ooh, interesting. Can you write it down?
Yes,
A
it's-
tumbling sponge.
Okay.
Because this I've never seen actually.
It,
I mean,
it looks
it
like
makes sense that sometimes they might detach because of currents, but I've never seen it.
This is
Yeah.
cool.
Because this happens in shallow waters. In the deep sea. Oh, look, another one.
How to get scientists excited, huh?
This
Tumbling
is
sponges.
super cool. Ah, look at that.
Oh. So this is exactly
Sorry, yes, this is
tumbleweed,
geeks talking.
Yes. Oh.
Honestly,
Yeah.
I don't think I've ever read that in the literature
Okay.
in the deep sea.
Okay.
Never seen that before.
I will make a note on the depth.
Yeah, yeah. If you can put the time and
Like on, guys. Didn't
I mean, the timestamp is there.
yeah, that's true.
I've typed tumbling sponge 3 times now. Just so we are
Is there a
certain.
way to have an idea of the current velocity, current speed, or
Speed,
yeah,
or whatever the ship is doing, but we're starting to stop them the whole time.
yeah, but just to know if this is actually what's the speed of the water or the
No,
current?
No,
Oh,
don't.
wait.
No.
We
Okay.
have no sensors on for that.
Yeah. Okay. Thank you.
Yeah. you move this then?
No, I touched it.
That was cool.
Yeah.
Frozen since you left.
Very special because it was two. And like,
Yeah.
I wouldn't
I
understand.
mean, one
Eh,
is already, but then you see a second one behind, and you're like,
what happened there, right? But
Yeah.
look what's coming up. Oh my
Okay.
God! We
Okay.
have another highlight there.
yes, I think you're in for a treat.
Mm-hmm.
We're at 968 meters. We're approaching an outcrop which seems to be covered with sponges.
Many.
So for all the sponge lovers out there, I'm sure you're many.
Yeah, nice. Yes.
This is maybe when the sponge garden starts, maybe not. We'll see.
Yeah. Wow.
Hmm.
Oh, is this Asconema foliatum?
I'm not sure.
Okay.
Can we stop before we ascend this part?
Where do you want to
Stop in front,
Oh.
so that we can maybe subsample one of these white sponges,
No worries.
because I'm not sure what they are.
Get one of them in my view on my camera. I'll know we're far enough.
Yeah, I think if you settle there, any of these ones would be good.
Okay, let's just come back a bit because that's high.
So I'm not really sure what this species is.
Which one exactly are you talking about?
The white
Ah.
fan-shaped,
the white
irregularly shaped as well one.
Mm.
But we're going to try to see if we can settle the ROV, and then we'll take a good look at them.
It might not be easy to slurp it, so
Yeah. I'm going to wait for you to... What is that?
Is that not
Is that an-
Craniella?
I don't know. Could it be a...
Anemone.
An-
Yeah. This is
anemone?
Craniella, right?
Yeah, this is
Yeah.
Camilla.
Yeah, I've logged that one.
There's lots of stuff to look at here. Look at that.
Wow. So many clams. Is it clams?
It is.
Wow.
It is. Okay, let's see what we can collect here or not, because a lot of things here are encrusting and
Asbestopluma.
Bridge.
Can
I-
you stop the movement
yeah.
for now?
yeah, it is.
Awesome.
Foliatum.
I have no idea what that sponge is. It's definitely a sponge.
come over on the way.
What is that?
Huh?
Hmm.
There's also clams in the picture.
Yeah.
Just a little side note.
No, that's true. That's true. Let's look at the rest, of course.
This is cool. Do you want lasers so we can measure it?
So the lasers
Ah,
are
they're
there.
there. Sorry,
Yeah.
I didn't see them.
Yeah.
Blind.
I know. So I'm getting texts from sponge lovers actually, who are also very excited about this.
Really?
I've never seen this before. As I said, I've been to the Schulz bank before, but no, this is different. Is this something that could be maybe collected with a vacuum, with a slurp?
1 sec. Yeah. Yes.
Yes.
Which tank do you want it?
So.
Sorry, there's
Like a
something else tumbling again.
yeah,
We have a lot of tumbling animals here.
a lot of current here.
Wow.
I mean, we are on the slope of a huge mountain.
We are.
we did expect some current, but
Okay, let's focus on the sampling event
yes.
first.
Would you mind that I
No,
that
go
I
ahead, go
continue
ahead.
to zoom a little bit so we have a bit of footage about the
just-
sponge?
You just go ahead.
There. Okay, the zoom is maximum.
Do you think we can group it with something else that we have collected?
Yes, we can absolutely do that.
We could even do this.
Well, I was told that there would be mucus from
That
the sea
is
star.
correct. That is correct.
So
Okay.
I think
Okay.
compartment number 3 would work.
Okay.
Do you want to? No,
Yes.
yeah.
I can do
Yeah,
that.
yeah,
You do this.
Yes.
The sampling part.
We can sample.
Okay.
Let's try with the slurp in compartment 3.
So I will make notes on the exact coordinates where we are sitting right now. So we have that for the species, for the specimen location.
Mm-hmm. So this is what we call an encrusting sponge. It's like a crust on the rock. Sponges for this are extremely important because they act as a cement between the substrate, and that substrate sometimes can be a coral reef. So they are actually very good to glue a coral reef altogether or other types of reefs. And we are going to use the slurp to try to sample these because encrusting sponges are very complicated to sample, but let's see. If you need to scrape a little bit the rock, maybe that will help.
So I start with the left one or the right?
So I only want one of the two,
Okay.
the whichever seems
Easier.
best.
That's quite hard.
I will add that we collected some of the rock
Yes.
animals. Would you mind?
No, not at all.
Thank you.
You might have gotten a little piece, but I'm not sure because I saw that you scraped a little piece, so maybe it came off. This is, you can see that collecting crust sponges in the deep sea is really, really challenging, and that's why they're so poorly known. For all I know, this could be a totally new species, undescribed to science. So it is important for us to know what it is, to know that it lives here.
And since we only sample a fraction of it,
Yeah.
Got it.
there's no harm done basically.
No, it will regrow.
Yeah.
I think we have a piece. Yeah, I think we have a piece. Thank you.
It may look a bit discreet, but
Yeah.
there's a lot of left.
Yeah,
now,
there's
right?
a lot of sponge left, so sponges can regenerate. That's the big advantage of having no organs, no tissue
Yeah.
to regenerate. It's super easy, and sponges do it very well. They heal in 1 or 2 days, and then they will just grow back. So, yeah. Do you want to look at something else while we're
I'm
posted?
good. I'm when they're ready to go, I think we can
Yeah.
continue the climb.
Sure. We are ready to go when you are.
Okay.
Just need to know where we are here.
Wow.
That is
It's-
it is really beautiful and
Yeah.
rich.
A lot of encrusting sponges.
Mm.
You can see the golf ball sponge here in the foreground in orange.
Bridge ROV.
RV bridge.
So we can resume movement, please.
full 330 continuous at point 1.
Thank you.
And now I see it everywhere. So it seems to be pretty common here.
Yeah.
And sometimes it makes those little bulges.
I just wanted to check one thing on one
Sure.
of the sheets, which is, you know how we always report the transects?
Mm-hmm. So we filled it here
Perfect.
when we
Okay.
started.
Awesome.
And I see we're still on T3, so
Okay, no,
Yeah.
no, all good then. Awesome. There's a sea star,
there is a sea star.
and it's feasting on something. You can always
How do
see-
you recognize that it's feasting? Every
Okay.
time you tell me this.
You see
So-
how it's let's say backside is
Yeah.
sort of rounded
Yeah,
and it's engulfing something,
okay.
because the way sea stars the way they feed is that what they do is that they have their stomach sac,
Mm-hmm.
and they actually how do you say, they push their stomach sac outside of the body like a big sweater,
Yeah.
and they would just engulf their prey,
Yeah.
and then they insert digestive enzymes, so they digest slowly their prey
Mm-hmm.
whilst having their stomach.
Outside
outside.
of themselves.
And first, when they're done digesting, they're retracting and they go back to a normal sea star kind of shape. But when they're feeding, they really, they're like in that position that we just saw.
Okay, okay.
Mm.
I'll try to spot the next one
Yes.
feeding on something. Thank you for explaining
Yeah,
that.
sure. Sea stars have they have such an interesting anatomy. I really love it.
Hmm?
They have gonads in their arms that look like.
So reproductive organs.
Oh yeah, sorry. Their reproductive organs are in their arms.
Yeah.
And they actually mate by rubbing their armpits.
What?
Yes, because the openings are in their armpits, so they're lying like this when they mate. And,
Wow. Okay, we have to find that.
Yeah, that would be amazing. And what else? They have like several canals inside
Mm-hmm.
through which they pump water, and then they have a lot of papillae on their back, let's
Yeah.
say. And when you zoom in, these are very beautiful structures. They have very sensitive tube feet.
Yeah,
lots of
just like
them.
sea urchins.
Yes,
Yeah.
exactly.
Mm.
So it's just, I love these animals. They're beautiful.
Haven't seen so many sea urchins actually here in the Arctic.
No, you're right.
I think we saw a couple, but
Not really, not very much. And you know what also surprises me? We have covered quite some ground with soft sedimented plains, and I at least haven't seen a single sea cucumber yet. And I really miss them.
yes,
I really
they
do.
are cute.
I want to see one.
So we are still ascending the Schulz Seamount in the Arctic on the western part of the Mid-Atlantic Ridge. This is probably the most famous or most studied seamount in the area. There's been a lot of scientific literature about the communities on this seamount, and yet still so many habitats and species to discover, because I mean, there's been many people that have come to this seamount in the last 10 years, but I'm still seeing new stuff. And we're still seeing even new habitats, which is quite amazing.
Sorry, I just need to have a little zoom there. Because there's lots going on. Look. Wow.
But it is extremely rich.
Wow. Wow. Hello, Anna. How was dinner?
Yeah.
So what time to go?
Bring it back in.
Yeah,
This
I'm
one.
back.
Back. Yeah, this one.
Oh, there's a tumbling Syrian again. Huh?
Uh-huh.
Really? It's
So it seems perfectly natural to have animals
tumbling.
detaching and tumbling now and then because of the current.
Oi, huge granite coming up. Wow. Canelas.
Yes. So I will probably need to sample one of those, but I'll wait to be on more flatter grounds, maybe at the summit. Yeah. Ooh, yes, that was nice.
Asbestos.
Yeah. Like this brown
Mm-hmm.
thing, I think that's a dead sponge. And when they die, they just become brown and settle on the ground, and their spicules just stay there. And they can form a new type of substrate for other organisms.
What are we looking at now?
We're looking at a crazy sponge. That's a glass sponge,
Okay.
probably Rosselidae as well, but it is beautiful. Looks like a hand.
Right.
Yeah.
Sitting on it.
Sorry?
Anna is back. We're back in amphipod with
Yes.
amphipod glasses, you know.
Yes. So after sponges, sea stars, amphipods, and tube worms. Look at this again. Lots of tube worms.
Mm. This is beautiful.
Mm.
Oh, wow.
Mhm.
They look so soft.
They are extremely soft, although they're really rich in glass,
Yeah.
and the skeleton is very complex, intricate. When they come up, when we bring them up on deck, they are extremely soft and fragile. They are very difficult to sample with the robot,
Mm.
but even more difficult if you use more destructive methods. So actually, the robot is still the best way to sample them.
Oh, soft corals. And what is this again?
This looks like Asconema foliatum, I think. This the flower.
The lace sponge.
Is that how you call it? Yeah. So you can see how delicate they are, because
Lollipop.
they're almost transparent.
And suddenly we have sea star.
Sea star.
And we have several species of sea star here, not only one.
What do you mean?
This is
Where?
I haven't
So
seen.
I see the big one. Ah, you mean
like,
altogether?
in the habitat
Yes.
that we have been exploring so
no.
far.
It's true. It's been very rich in sea stars.
The richest so far.
Mhm.
And I love that the soft corals are back.
So we are now at 940 meters.
Right.
The communities seem to change a little bit. With
Mm.
you can see more and more of these really large vase-shaped sponges, the white vases. These are glass sponges. Mm. Do you know what that is?
No.
No, oh no, maybe that's actually a carnivorous
Yeah,
sponge.
I think so.
Yeah.
I think so. Shall we hang in front of the soft corals, like a little climb, and
Line a
then?
little bit further,
Yeah,
and then
yeah.
Yeah.
I agree.
Yeah.
And then we will look for amphipods.
I'm not any amphipods.
No.
I'm
No.
looking for
We will look for
small
small things.
So Rob,
Yeah.
there is something very beautiful coming up. Like if we climb, like if we could just hang here for a second. So
Yeah.
Here or just a little bit higher?
what do you think, guys? A bit further up, and then
Bridge RV.
RV bridge.
Can we stop for a moment, please?
I'll stop.
Thank you.
so maybe we do, if we can, climb just a tiny bit up, and then
Stabilize.
and then we still, and then
Mhm.
we can zoom.
So I wouldn't be surprised if the brown texture we're seeing is maybe dead sponges. So like what we call a spicule mat. Spicule is the name of the skeleton of sponges, and it creates this rather thick and coarse mat on which other organisms can start to grow.
How about we zoom in on this little elevation there?
Absolutely.
Wherever you can place it, but most stable.
It's quite solid here, so we can rest against it.
Okay, yeah, I think we would like to have a still of this.
So if I would come back a little bit,
No,
Yeah.
no. Oh, yeah.
so you get an overall view.
Maybe I wait for us to be
Yeah,
stabilized,
Yeah.
and then I will zoom.
Wherever we can stabilize.
yes,
Sure.
exactly.
Yeah. So you have to imagine that this robot, this remote operated vehicle, a ROV, is more than 3 meters long, weighs more than a ton. So it's a really large piece of equipment. Lots of technology in it, lots of cameras, wires, computers. And it is challenging to place it when we want to make observations, of course, because you need
Which
Certain-
area there? Which area?
flat
Which area do you want?
substrates.
Anywhere where you're stable.
But yeah,
To the left, a little bit higher.
and the thing is that we have a screen right in front of us and we see something that is very interesting to our eye. But then the pilot flies by a different screen because the pilot has a completely different focus on than what we have.
And then they have current.
Exactly.
and,
So
yeah.
the communication,
And it's really, look at the Assyrians.
yeah, amazing. This is really cool.
Thank you. Thank you. Okay.
Sorry.
What is it? Oh. Yes. Yeah. Are we good? Or you want us to?
No, that looks good.
Looks good.
Okay.
Thank you. So I'm gonna zoom in a little bit on this coarse mat. You can see the golf ball sponges quite well.
Wow.
And all the bigger stars, yeah.
All the brittle stars. I'm not actually sure what. Are these old maybe tube worms again?
Can we zoom in more or are we
Sure.
maxed?
I think I can zoom in a bit more.
This is very interesting.
Looks like old tubes or some kind of tube.
But
Satellites.
it could be that they're the old version of this white one
Yeah,
that we see around,
yeah,
because it has a different structure
yeah.
than the
And are these
polychaetes before.
bryozoans? I'm not entirely sure. They're
I've
not
not
sponges.
Then it's likely that these are bryozoans.
Quite long to be a bryozoan.
But
Yeah, the shape is yeah.
they resemble something that I've seen in our catalog. That was this is the Norwegian Deep Sea Image Catalog by Riley, Meyer, and Irina and Rosalind Fredrickson, Irina Zule and Rosalind Fredrickson. That is it's basically a catalog that is updated as we speak, basically, because we will deliver more species names, especially for the sponges.
Yes.
And I think that I have seen. Look at this.
Right.
Yeah, it's only bryozoa in that, but like
Yeah.
I think this looks very similar.
Yeah, but it looks more regular here.
It does. It
Here
does.
it's more coral-like in a way.
Yeah, yeah.
You see what I mean?
Yeah.
So I don't know. But I think actually when we slurped at some point the encrusting sponge, we got some of that.
Really?
We did.
Okay.
So it's in one of the compartments. We will have a piece.
A little bit on the left, just a little bit.
Sure.
Staying in the sea.
There's another golf ball sponge.
I think we were looking at two different species of brittle stars.
Yeah.
lots of different sponges as well.
You see the tentacles of this worm on the right side? Of
Here.
the- of the
Yeah.
substrate, of the
Yes,
substrate.
absolutely. There's a worm that's outside
So if you look very closely
on the right.
on the right side of your screen, a bit lower than the center, you see a white tube. And in the end of that white tube, you have a
crown of tentacles.
crown of feather-shaped tentacles.
Mhm.
And these are the bristles of a worm. Really beautiful. Extremely rich. Just this little spot. I mean,
Yeah.
we
yeah.
were looking at like 15 centimeters of seafloor, and we see all this. It's really top, really, really cool. Wow.
There's a group of friends here chatting.
Do you think they're having a little tea party?
Yeah. This is Asconema mega atrialia. This is also a glass sponge, and here you have 3 or 4 clumped together. And then other sponges, ascidians, tube worms. Yeah, it's very rich.
Beautiful shot.
And it's funny because just after there's like a strict border, and then you're in another type of habitat with none of these tube worms.
It's very steep. Maybe there has been
Yeah,
a landslide.
exactly. It looks, yeah, I agree. It looks like a landslide of some sort. Or, yeah.
Shall we continue the move, or what do you think, guys?
Yeah, it's a bit far away.
I think just go to the closer to the left as your they come up. Just down. And then one more zoom there, please.
Here,
just
here?
yeah, just where you
Okay.
are. Stop. Very nice.
It's nice, yeah. I just want to take a look at that one. While I'm there.
Yes.
I'm not sure what this is. Mm-hmm.
Okay.
Yes.
All right.
Thank you.
Bye.
Bridge, ROV.
RV bridge.
Yeah, continue the move, please. 330 at
What's
point 1.
That's what I was going to suggest. That's what I was going to suggest.
Yeah.
If you want to,
Find either way.
yeah. Do you want to take a pause or?
I can take a little pause, and then we'll just.
Or just copy.
Yeah, we'll just. Yeah, we'll just swap,
Yeah.
and
Sure.
as we always do, us three just hanging out here.
It's nice to meet you.
Yes. I'll be right back. Do you need anything?
Can you just pass me my water just behind?
That one?
We've been talking for so long. Thank you.
Yeah, I also need to. Are you guys good?
We are
Yeah,
good.
no, we're fine.
Thank you.
Well, I am.
Yes.
Yeah, we're good.
Some more lollipop sponges.
Can actually get the tape and bring it up.
Oh yes.
See you soon, guys.
Yep. So we have been sampling a little bit more.
So I
One
saw.
lollipop sponge and one encrusting sponge, but then we took actually other things as well, so we might have.
I saw it, but via the suction tube as well.
Yeah. So we are at 930 meters, still ascending the Schulz Bank Seamount, close to the Arctic, mid-Atlantic ridge, during the deep Arctic expedition. This is our first day at this site. Here are some golf ball sponges. Sea stars. Yeah, we have regular places where we find these masses of tubes, or it's very strange.
Or tube cemetery.
Hmm?
They're built of calcium carbonate. So they're very sensitive to ocean acidification.
Why is that? Can you explain a little bit what you mean by that?
Well, the calcium carbonate pulp is destroyed when it from acidic, same when cleaning the bathroom with some lemon.
Mm-hmm.
And it will dissolve, and it will not. That will affect a lot of animals. So it will not immediately dissolve them. To have thinner shells, then they're not as robust, not as stable to protect them to predators. It will shift entire ecosystems.
Yeah.
from-
Absolutely, and they're not the only animals with carbonate calcium.
Of course not.
So it will be an issue for shells, clams, maybe even for calcareous sponges, because some sponges also have carbonate calcium skeletons. So So we're soon going to reach the 900-meter limit on the Seamount. I should remind you that the top of the Seamount is at 600 meters, so we are about 300 meters from the summit.
So are we close to the end of our third transect?
I don't know.
Can you zoom out on the global map for that?
Let's see on the map. A bit more than halfway. Yeah,
Okay.
yeah. Thank you. So this white crust sponge that you see, this is the one we tried to collect earlier. So I don't have a name for it. I'm very curious.
Is it similar to the one that we had before with
I don't think
like
I've
an
s-
encrusting?
no, I think we haven't seen it before. At least I haven't seen it before.
But it also has this greenish shimmering.
Yes, this one you mean, like
No?
No?
I meant the encrusting one. It's not.
the white one?
No, that I mistook for an Ascidian first. Like really small.
Uh-huh. Show it to me when.
Well, when it comes up
Yeah.
again, I will.
Here's a beautiful glass sponge in front of us. And we are hitting a wall. Wow. Looks very steep. But there's still some lollipop sponges, soft
soft
coral.
corals.
But this is a massive wall cliff. Again, a Roselidae sponge, one of these glass sponges in shape of a vase. Sea stars.
Stop.
Can we stop?
No, sorry, we missed it. Sorry, we're too late. I wanted to look underneath.
Underneath this?
Yeah.
Can we go back down 1 meter or so?
Yeah.
Maybe I can look down.
It's fine. It's
It's
fine,
fine?
to continue.
Okay.
Yeah.
It's good?
It's fine to continue. Yes.
I thought it's coming out there, but
Ah.
on it.
Mhm. Do you think we can try to sample this one? Okay, then let's try to settle, and maybe we can also grab a few other things around this one.
show him on that.
Just in front of that.
To get that white one.
Too far.
Sorry.
Too far.
No, I said just show him because he wasn't, because he didn't have it on his screen.
Mm.
And I knew.
We're going to try to see if we can stabilize ourselves on that slope. And then we will do a bit of zooming to look a little bit at the small things.
Great, Charlie. Can we pause the move, please? Thank you. You want to slurp this?
Yes, first we're going to make some
Yeah.
stills.
Photos. Yeah.
Yeah, we will tell you when we can sample. But
Lasers.
like this it will be good because we will have
Lasers off, please. Thank you.
at the
Can
same
go
time.
back on again. Can I? No, do you mind just
No,
letting it?
go ahead.
Because we can see all the
Sure.
Bertle stars. We can see the anemone. We can see the clam, the Ascidian. We can see the polychaetes, the rhizome to the left. So we can see a lot of animals surrounding a sponge. Now we're zooming in on having a sponge in the middle.
Old-fashioned.
Some brittle stars buried in the sediment.
Mhm.
I will actually write the time. So we are 73 degrees north. And we are at 908 meters deep. Ah. Yes, that's a tubeworm.
I'm not convinced. We can also move the entire thing to the left, so we can also look at the anemone.
Yeah.
Yeah. And then you can see the mouth of the anemone right
Yeah,
in the middle,
that's-
the little ring.
Yeah.
It's opening.
So you have a
The
sponge
feet.
on the right and the anemone on the left. Mm. Should we zoom out a bit more?
Yes.
Are you ready?
We'll zoom out and we're ready to sample. But I would no. Yes, I'm ready.
Yes, we are ready.
Okay.
So just the white one.
Yes, and you can put it in compartment 4. Which is currently empty, but because it's a complicata, I don't want to mix them. But otherwise, we have been putting different things.
Do you want us to zoom in more?
No, that's fine.
So we are again using the slurp, which is quite efficient to get these sponges.
You're spot on. Good. You're absolutely on.
Whoa, perfect, perfect. Yes, and
Can you
you.
see the tube? Is it
Yep.
all gone through?
We have to zoom out maybe to see the
Yeah,
whole
no,
tube.
you see it on the other screen. We can't get so far.
Yes, look, because last time we had a sponge that was there.
Yes, that is true. But I
I've
meant
seen the
egg-
tank.
Yes, that's what I
Ah.
wanted to see.
Ah,
Thank you.
sorry.
You can see
tank.
that's that big.
Yep, perfect.
Yeah.
You've seen it down to the bottom.
Perfect.
So right, okay.
Absolutely right.
And we even got a little bit of these dead tubes, so
Mhm.
that'll be interesting to examine a little bit the substrate. And we even left, I don't know if you can see it, but we even left a piece of the sponge actually, so it will regrow. You can see here.
That is fantastic.
So great job, Rob.
Yeah, thanks a lot.
No, team.
Do you want to something else?
I
Or
would
should
no,
we-
just like I would just like to look just at these two little things, and then we can. Oops, sorry.
This is cast
I'm so
8.
You're not excited about this one?
Oh,
Mm.
oh, it was just below?
Well, I'm not telling you because I want to see this one.
Oh wow, yeah. That's a little shrimp.
It is, but look what's poked there. And there.
Oh wow.
Very nice. I will tilt this if you don't tilt as much
Yeah.
as you can, just to here. And you see the clam here. And on the right side
Yeah.
is again one of your
Yes.
the nice sponges
Yes, absolutely.
with a birdle star sitting on it. And you can also see really there's lots and lots of birdle stars on the seafloor. As I mentioned before, they've been counted up to 1,000 per square meter. 1 meter, more than 1,000 animals. It's a huge amount of biomass.
Mm-hmm.
Also changing the seafloor. You see the little anemone in the front?
So maybe we could also collect.
Ah.
Now that I've seen, no, because I don't know what this is.
I knew it. I knew you would want that.
And but we've been seeing it a lot, and I've been restraining myself, but now I have to say it. I don't know what
Okay.
this is.
Just for Jenny, we'll move over to this to the bulk.
Yeah.
There's
It's
a little carnivore sponge here. As
Yeah,
well.
no, I'll ignore that for now, and I'll show Jenny the bulk. Yeah, and you know our snail on the left.
A snail. Oh yes.
Hey,
Oh,
come on.
nice. Very nice. No, but I really like this guy actually. It's beautiful. It is.
Which one? The one
This one.
poking out of the.
Yeah.
I can't see. Okay, we're looking here. Well, it's
Interesting.
very hard to see for the untrained eye, I guess. But I don't want to be so annoying with my amphipods, but there are two little ones there, and there is also one on the top.
So where exactly?
They
Are
have
you
the
looking
shimmering eye, just there. But
Oh, under
yeah,
the
but
rocks. Yeah.
it's- I was just interested to see. And where do you want to go, Paco? You want to take over? So
Yes.
You want to,
So if we can, yes, let's try to select that one. Oh. And we can put
Just
it
right
in
there.
the same
fork.
fork.
Just right there.
And with a slurp as well.
Sorry, you want slurp it?
Yep.
Number 4.
Yes.
Right.
The brownish one.
Yeah.
The lower. You want to select the lower one?
Yes, this one.
It's right there.
Whoop. So,
Yeah.
Yeah. That's right.
yeah, did you get a piece?
Yeah.
I think
Oh,
fantastic.
yes, you did. You did.
That was fantastic.
Yes.
That was just
Surgical, because now you
Yeah.
have left some, they would regrow,
They will.
so beautifully done. Thank you.
Thanks a lot.
That's right.
Yeah.
Well, your head's just disintegrated.
Sorry?
The head's just disintegrated. You can see all the bits.
Yeah, but I mean, even if it's in little pieces,
Yeah,
I will manage to get some DNA,
There's some big
some
bits in there.
skeleton. Yeah.
And we've got all the beautiful images
Yeah.
before having a
Yeah,
piece.
exactly.
This is all good. Thank you.
Yeah.
We can continue. This is-
Yes. You finished there?
Yes.
Right.
Thank you.
Great, Jeremy. Can we continue the move, please? Thank you.
Well, you see,
So-
it's
some-
everywhere.
yeah,
This brown.
sometimes this rock has this shimmering color that I think they're encrusted by an obsidian, but it's a trick on the eye because of the light.
Okay.
So these are rocks that aren't covered by sediment. Come. The next time we see so many holes coming up, like here, but let's continue for now. But the next
Mhm.
time, let's see
You
ahead
wanna
of time that we
stop?
would stop. Yes,
Okay.
please. We have this more soft crumbles.
Is Paddy there or Rory there? Anyone, please?
Annie's on his way.
Okay, thanks. Let's see.
Here we have two habitats here. One on the right with the tubeworms, one on the left with rock, and not much growing on it.
Now we have the pink anemones coming back, and the ascidians.
Okay.
But it definitely seems richer on the part with a mat made of tubeworms and mix of tubeworms and dead animals than on the rock.
Also, you see all the Birdle stars when we zoomed in. They also form quite a substrate because when they fall apart,
Mhm.
these are quite
Oh,
hard little
yeah.
particles.
I hadn't thought of that. Yes. Mm. So the remains of all these animals, or at least many of these animals, is really important to create a substrate for
Yes,
the other animals.
yes, it's essential for their larvae to settle.
Mm. It's like they're all contributing to their own environment and habitat. Okay, we have now passed 900 meters. We are at 897 meters, about 300 meters from the summit of the Schulz Seamount, on the western side of the Mid-Atlantic Ridge.
Oh, that's especially pretty anemone.
Yeah. Again, we have more and more of these Rossellidae sponges, glass sponges. Super bright white.
And soft corals. And a shrimp.
Mm-hmm.
And a feather star. And a glass sponge. And more shrimp. More anemones. More soft corals.
All in all, very rich habitats. Mm. Here's maybe a geodia.
Can we stop, please?
Anywhere in particular? or-
No. But just to have a focus here. Maybe by the coral and the sponger, a bit ahead of us. Just
Is this for review or?
just for viewing. Just so that we can zoom in and not get car sick. Thank you. The lollipops
Yeah.
on the left.
Reg, can we pause the move there, please?
Thank you.
Lovely job, thanks.
Now, look at this. You'll be amazed. If I'm not mistaken completely. Can't see. No, I cannot recognize anything. Sorry.
That's okay. Maybe we can still
Well,
look for.
Yeah, well, looking for nice critters and whatever we can see here.
The soft coral is amazing. It's beautiful.
It is.
And the sponges.
Are these two different soft corals here?
I don't know, but
I
this
think that-
definitely looks different from what we've seen so far. Or maybe it's because
but
it's
also
more.
this one and
yeah.
also this one, they definitely look
Yeah.
different.
Can you actually zoom on those polyps? Do amphipods live on soft coral as well?
Yes,
They liked?
Yes.
Okay.
yes, for sure. We haven't sampled any corals yet, but I
Mhm.
have picked part of corals
Mm-hmm.
for amphipods, and there were huge numbers of very small. You see, actually, after you tease me with all my amphipods, I've gone a bit shy, but I can point out this amphipod sitting on the very lower branch of this soft coral.
I see it.
Thank you.
Yes, I see it. So if you look at the bright pink soft coral, one of the lower branches has a little amphipod sitting on it. It's kind of whitish,
It's
big eyes.
well, there
It's
are-
moving a lot.
Sorry, there are actually 6 more in the inside. You can see them just. I can just see the eye. It's really not that obvious.
Okay.
So I didn't want to point
in-
them out.
In the transparent one here?
No, in
No.
that one. You can
Oh,
see
in the
here.
bright one.
Yeah.
Okay. Okay.
We're a bit far off for those. Well, amphipods often they like to hide. They also sit in the large sponges, as you know,
Mm-hmm.
but also in any kind of corals, especially also in cold water corals. And
So how is this scene different from New Zealand? Because I know that you've been working in New Zealand a lot. How is this different, for instance? Would you see this type of scenery as well?
yes, you would. Just, I mean, it's how long is a piece of string kind of question. Only in our last few days here, we've seen so many different habitats.
Mm-hmm.
Like,
But
like
like
our
would
bamboo
you,
coral.
Yeah.
I mean, it's not typical for it.
yeah.
And what I'm trying to say, there are so many different habitats in New Zealand. There is
Yeah.
not one.
No.
You've got the deep sea trenches, the corals, the troughs, the
Okay.
canyons.
But in terms of dominant
the
species,
the sponge grounds.
yeah.
And we
Mhm.
also have areas with lots of sponges and very beautiful cold water corals.
Mm-hmm.
I had the luxury to join an expedition last year, which was also fantastic. But let's focus on this year now. It's- You see the shrimp?
Yes. No, I do.
Very good. And now what's this? Oh. There's several brittle stars. Sometimes they're half buried, so you can only see the legs, but since there are quite a few of rather similar sizes, you can be sure that it's a brittle star, where the
Mm.
leg is poking out. Oh, what's this shell do?
So here we have 2 sponges. This one, I'm not quite sure what this is. But the one just above, that's the woolly sponge. That's what I've been calling the woolly sponge because it looks very hairy. And this one is Hemigilius arcofer. And then there's a little yellow keratose sponge on the side. I actually wonder, could this be an Assyrian or it's a sponge? You know, we've been seeing these white spots a little bit everywhere, but I cannot.
Which one? You mean the substrate
Yeah.
of the?
No, the white.
I think it's.
It looks to me like a sponge, but I know that
Yeah.
sometimes they can be confused with Assyrians. So.
Looks like sponge to
Mhm.
me. This one is hiding from us. It's running off.
Yeah.
See, see them what? They put their arms to the front, hold on, and then pull their body back. Do you want to zoom somewhere else, or should we continue?
We can continue.
Maybe one lower to the right.
There's a nice feather star. Here again we have a lot of tubeworms. Serpulidae.
So. Ready to go?
Ready to go.
Thanks a lot.
Oh, here's again your colorful. Was that a crinoid? Or no, that was a sea spider.
That's a
Can we
sea
continue
spider.
to move, please?
Sea
Yes,
spider.
we continue flying.
Maybe
Sorry.
log it, this one,
Yeah,
because it's
sorry.
really special.
Thank you.
So we're approaching, I think, the end of transects.
Yes. Very nice.
So we're not.
Is that an urchin shell?
Yeah, that's probably the first clue that sea urchins are around. We haven't seen one alive yet. They're definitely there. There's the pompon sponge again. The fish and the lollipop sponges. We're still about 230 meters from the summit on the Schulz Bank Seamount. This is a very, very large mountain, about 2,000 meters high.
Now, shall we stop again when we get back? I'm going to look at it. Or maybe zoom out.
Maybe we're very close.
We're just zoomed out completely.
Yeah, yeah, yeah.
It's strange.
Because I
We
think
can
we're
zoom in here.
very close. Ooh, this is nice.
Mm.
A lot of soft coral in this patch here.
Dry lab, can you hear us? Do you want the sexton out? No worries. No, they don't. So let us know.
A lot of golf ball sponges here. All these orange dots are little golf ball sponges. And more of these tube reef walls that we started to see at the beginning of the dive. So they continue to appear now and then. Mm.
We're now 860 meters. And more soft corals.
Yeah. Can we start to sample this one coming your way?
We'll try and sample again.
With the slurp, right?
Yeah, we can continue because it works really well with that species, the slurp.
And this kind of sediment too.
Yeah. So it's this one. Yeah. So this would be
Cast 9.
cast 9.
If this was the movement. That bridge RV, can we stop the move there, please? Thank you.
Can we have a look? Do you want the lasers on first?
Sure, we can have
Each tank.
it will be the last one.
4.
It's 5 because there's something else in 4. Yeah, 5.
Okay.
Could we have the lasers for size? Thank you.
And off again.
And off now for zooming in.
Thank you.
Thank you.
So you have the to be sampled sponge straight in the middle.
Yeah.
You've got 5 anemones. Yeah, distributed above. Soft coral below. An ascidian. A clam.
A golf
Zoom
ball
in.
sponge,
So 3 seconds.
bottom right.
Yeah, but now we'll zoom in. We've seen Ascidian. Yeah, and you see crinoid. That's,
Yeah.
no, look, there's also
Yeah, I was thinking it's nice.
one living in.
Oh, is that a brittle star living inside the sponge?
I'm not sure, I assume so.
Yeah.
I assume so because there's also one right to the right of it. When your eye gets used to the image, try and spot all the Birdle stars on top, here on above the sponge on the sediment. You have this slightly purple disc and this slight rosé colored legs. There are quite a few.
Yeah.
And a little bit down.
Sure. A bit more.
Also the sponge, but I think it's, we'll zoom in whatever we can here while we're so stable. That's our maximum. You can see that it's got like two parts to it.
Absolutely, yes. It has a like a skin of some sort in which the water circulates. Actually, that's the canals that go inside the sponge and bring water to the whole sponge. And yeah, it's like a detached skin, somewhat. And also something we've discovered is that in the specimens we collected, several of them were reproducing. So we know absolutely nothing about the reproduction of that species. It's the first time we try to study it, and it's actually reproducing right now in some specimens. So we don't even know if there's male and females or if they're hermaphrodite. This is something that we will investigate.
What's a hermaphrodite?
So hermaphrodite would mean that the sponge would be at the same time male and female.
Which is true for all animals that are hermaphrodites
Yes.
and always have both genders. Are we ready to sample?
Yeah.
Mhm. Look at this little I think it's a cucumber actually. To our bottom left. It's tiny.
Oh wow. Yes, very small.
Mhm.
So, could you zoom out a little bit more? I want to show that there's also a craniola that you could get after the same range. Zoom a little bit more. It's there. This one. If it's not too close. Yeah, this one.
Do you want to get that first, or?
No, we can go with the white bush first, and then exactly, it's this one.
Oh.
Do you want us to zoom in, or is that
That's
fine?
fine.
That's fine.
Yeah.
Do you actually mind holding on?
Wait.
Just holding on while we. I want to see something. I want
Do you want
to.
to move?
Well,
Yeah.
excuse me. We'll go back to your sponge immediately.
Yeah, sure.
But I want to zoom on this little hole. This, yeah.
I can help you to
Thank you.
put it in the
Yeah,
center.
thank you. We'll focus on the sponge. Oh, sorry.
Yeah, we're ready. Thank you. So we don't need to get all of it. If we get a piece, we're happy.
Okay, I'll just get to the side of it.
Yeah. Perfect. Perfect.
Absolutely
if it
fantastic.
arrived in the box.
Yep, yep.
Yeah,
Fantastic.
absolutely perfect. And
Can the golf ball go into the same box?
they can do in the same box, no problem there.
Yeah.
And that will be cast 9.
Yep.
Perfect.
And we have departure.
Okay.
Thanks so
Thank
much.
you. Is there something else you'd be interested to collect?
No, not to collect.
No?
Just have a little look first.
Yeah?
Hmm? You see another well? Right in the middle?
Yes, now I see it. They're really hard to spot, these little snails. So what you also have to know is that all the sediments that we collect when, for instance, we slurp a sponge, the sediment will be sieved, actually. So by you and Jenny, you will, and then Helena, you will look at also the little animals that are living in these sediments. And sometimes you find really nice
Oh yeah,
specimens.
quite often.
Yeah. So
But it's impossible to see these with this magnification.
Mm.
But there's lots of little critters living amongst it.
So all the
Saint-Pons.
sediment that is vacuumed will be sorted and sieved
Yeah.
on deck as well.
Our mesh size is 1 millimeter.
That's the smallest mesh size.
No, we've got lots smaller. But for this kind of sampling and for the animals that we're interested in, and also that Solvin here on board can take pictures.
So you
Yeah, so
retain
anything-
everything that's bigger than 1 millimeter,
Yes,
basically.
yes. Even so, some of them are smaller, but they could have gone through.
Mm.
But if you make the mesh size much smaller, then it's very hard to sieve because this is a muddy substrate. It doesn't look as muddy, but it is quite dense.
So it sticks.
It sticks together, and then
Mm.
you destroy everything when
Mm.
sieving. So, buy bulk, buy operates. Let's continue our climb up.
Yeah.
Thanks
we are
a
ready.
lot.
Thank you.
Bridge RV, can we
Which
continue
slope
the
was
move,
that in?
please?
5.
Taking you.
Cast 9?
Slurp 5. Cast 9. Yes, cast 9.
Okay.
So unless we really take different things, now they are quite full, I would say.
Mm. What
But
about the mugs?
the bio boxes are entirely free.
No, the mugs.
Oh, the mugs, they're there. They're also
the-
free.
Okay.
Well, at least the number 2 is free. The other one will be maybe a bit
Yeah, of
challenging
course.
to take
Yes.
out. Possible, but not entirely sure. These
So
interesting, yeah, encrusting sponges are really strange. I'm very curious to look at the specimen we sampled when the ROV will come back. We are 2 teams on board working on 2 different shifts. So 3 scientists working from 2 a.m. To 2 p.m. and 3 other scientists working from 2 p.m. to 2 a.m. And like this we work 24 hours. 24 hours. Yes. Jenny,
Hi.
you're going to replace me
Yes, I will.
for a little while.
Sorry, I got stuck in the lab.
No, no
And
worries.
then time flew by.
Enjoy.
Thank you. Hi.
Hi. Do you mind swapping places?
No, not at all.
It's mainly for temperature.
Yeah.
Because I'm sitting directly
know,
under this
that's-
air condition, and it's there's a cold spell underneath. Thank you.
Okay. Back in the seat here. You know, we had we had a sample in the lab that I really wanted to look at, and then
Which one?
sea star. And then we had some visitors in the lab, and then it got chatty, so time just flies.
It's because everyone on board is so nice.
Yes.
I'm not saying that to be polite while being overheard. It is really an amazing expedition so far.
Oh, we were almost at the end of the
Yes.
transect. I see.
Yeah.
Okay. It's so beautiful.
We still haven't used the mug, so
Ah.
maybe we wait until the next transect
Yeah.
because our slopes are quite full.
Yes. That's true. Oh, how was the sponge called again? Ah.
I don't want to say the wrong names. I'm guessing myself.
I think it was Asconema foliatum.
We have it.
I think so. I will log it. I dare.
Brave girl.
I am very brave. We have lots of potato ascidians there.
And the same anemones again, and soft corals.
Yes.
And lots of little rocks. And the nascent sea urchin shell.
Oh. They must be high, like, they must be
Yeah.
quite camouflaged because we always just see their white shells and there must be living urchins.
Yeah, and also sea cucumbers. I'm still waiting to.
Yeah. It's funny that you mentioned this because when you were not in the shack, I told Paco that I'm really missing them. Like,
Me too.
especially from the soft sedimented plains that we've been flying across. Not a single sea cucumber. I mean, where are they?
But we've got little stars here instead.
Now we have like, yeah, on this side. I brought the motivas.
That's.
You've seen them.
Hmm? Sorry?
Yeah, that's
See.
good. That's good.
Well, it's a tradition of this in our working group to bring this one big sampling bottle pot full of little sweets, and we keep on restacking it. And so Jenny now brought it here for our pilots.
Thank you, Jenny.
Cheers.
Even has a label, Motivasa.
I mean, we need a bit of late night snack here in the shack, huh?
Oh, is it a cucumber?
No, it's an Ascidian tricking
It
you.
is. It is.
What is this?
Soft coral.
It is.
Yep.
It looks so sort of dense.
It's true, but those of course.
Okay, okay. Are the lasers on? Thank you. That's Homatida. I will note that down because now I see it. Homatida anemone. Now it makes sense. I have been thinking about what kind of anemones they could be, and Homatida, of course. Yeah. And Actinostolida. So the big pink one we had on this amazing boulder earlier, that was Actinostolida.
Shall we stop again?
We can. Anytime.
Do you mind stopping again? Thank you.
Here or
Yeah.
anywhere?
I
Yeah.
don't have no web specific wherever suits me.
What's
I
that?
would just like to zoom in. That's
What is that?
as soon as we are stable, I will zoom.
And is that just a sponge? Now I'm seeing things. Wow.
Bridge RV, can we come to a stop there, please?
Yeah, we'll just
This is a
start
colonial
circling
Assyrian. I really would like a zoom.
I'm nearly
Thank
zoomed
you.
out. We will. Can we turn off the lasers, please? Thank you.
Yes.
So just having a close look at the soft corals and the anemones. Again, brittle stars. And on the very left, you see the Assyrian. Right. And we'll move our camera there now.
Thank you.
And
Oi.
Where you going?
Are we zoomed in all the way?
now we're getting there.
Wow.
Now count your offers.
Yeah, count the little stars. I think this is so we have a sponge, and by the way, I got a notification by our sponge expert Julio. Where's my phone? It was just here. There it is. And he actually texted about this encrusting sponge, and he said that's a forsythia, the one we sampled, and it really resembles that. So I will just make a little note. And we have a colonial Assyrian in the top left.
Yes.
Is that an amphipod?
Right. It has to move to confirm what I'm doing.
I think so.
I'm trying too hard to look at other things here.
Ah, it is an amphipod.
I know. I know.
It's obvious. We have
It's a
clams.
family Calliope.
Yeah.
It is.
I will actually make a note. Calliope.
We can
Clams.
So I'll show you. Oops. If I would. What am I doing here?
Sometimes I wonder if people actually see what we're seeing, given that you really need a trained eye to see these little critters.
Oh, but they see maybe. other
is-
interesting things like rocks, the different rock formations and
Yeah.
colors and minerals and sediment that we can't really distinguish. Don't worry, just empty it.
Just-
plenty to we will refill it. No worries.
Our pilots are really having a little snack there while we're
Well,
We're having a meeting. What is
you
that?
can have a break
actually.
when we're
I wouldn't mind to have a little snack myself, to be honest.
Do you want a bit?
Yeah, I think so.
You can have one, so.
You have
Okay,
one, yeah.
okay, okay.
Yeah, go that way.
Thank you. Look at this huge jar. Do you also want something?
No, thanks.
You're good.
Nice, thank you, Jenny.
Cheers. This is like watching TV. Slow TV. Really slow TV. Rob, just take it back. Keep it in a safe space.
No?
Trouble is I can't follow the storyline.
The storyline.
It's moving.
Our storyline is very slow.
Mm.
Zooming very slowly.
It's much easier on the eye when you zoom it slowly.
I think so too. You're looking for amphipods.
No, no. Who gave you that idea? Come on.
We can continue.
I don't point them all out otherwise.
No, let's go.
Reputation here.
Let's finish the climb.
Yes, we will.
So we are on
We're
the
zooming
on
back.
The oh, what is that? No, it's just an enemy. Never mind. All good.
So we can continue, please.
Yes.
Thanks a lot.
Another Ascanema foliatum. Versalida. Wow, lots of soft coral. Wow. It's really colorful. There was one more Colossandeus, Proboscidea, actually. I saw it on one of the screens inside.
Yes, we saw it as well.
Got really excited.
Yes.
Really nice. Ah, speaking of which,
You see?
can we?
Yes.
Sorry, can we hold? Just
don't
hold.
Don't zoom in.
I'm not. I'm not.
Thank you. Okay, this is a very funny coincidence. I was just talking about a species of sea spider that we had been seeing on this dive twice already. And just as we speak, we found another one. It has the beautiful name Colossandeus proboscidea, and it is quite a large sea spider.
This thing here.
Yes.
Yes, Rob, that's correct. No.
Nope.
Are you
It's this
seeing
one.
it?
No, I can't see it on your screen actually. Which is really strange. In ours, it's centered.
Yeah. That's what what's in this one. It's just there.
Okay.
Yeah. Yeah. And are the lasers on or off? Sometimes I don't know.
They're on.
Now they're
Thank
on. So the lasers are 20 centimeters apart. And now the sea spider, we have to zoom in on it
Yeah, we
shortly.
have to sit first or stay stable first.
I agree. I'm just excited because it's such a huge animal.
Yeah. Or say there. Yeah. What we'll do is. Excuse me.
Oh, bless you.
Thank you. I'm going to try and sit down here so you can get a stable picture.
That is super nice of you. Thank you.
Could If you scooch a bit towards stop, no, maybe not. No.
Let them sit first and we can.
Yeah. Yeah.
Okay.
It is very tricky to sit here even.
Perfect.
Wow, now it's right in the center. Amazing.
Slowly edging.
Wow.
So colorful.
So beautiful.
Where is it made?
Wow.
Thanks a lot.
amazing.
Okay, go for it.
Wow, what an image. We will just measure it once with the lasers and then we can turn them off for the beauty shots. So this sea spider is more than 20 centimeters large.
Lasers off, please.
Thank you. Ah, this is amazing. Wow. Oh, I love this. Our good friend and colleague Caroline Ulia, she's currently doing her PhD at the Senckenberg Institute in Hamburg. And one of her animals of study are those sea spiders or pycnogonids from the Arctic. And she will be absolutely fascinated by this footage because it's so rare to come here with an ROV to sit down on the bottom of the seafloor at 833 meters depth and just getting all these nice shots of these animals, and now in particular this beautiful sea spider.
It is super large.
It's incredible, really. Wow. I need to take a picture.
I adjusted as well. And also the anemone on the left.
Oh my god. Okay, Caro, I hope you're watching. This is just so cool. Can you please move? That would be amazing.
So it's starting.
There
It's
we
starting to
go.
move.
What does it have there? What's dangling below? What's happening? Do you see what I'm seeing?
Yes, of course. And I wonder if that's another crinoid underneath, having the arms out.
What?
Do you see?
What?
Do you see what I'm getting?
No.
There's another one underneath that we're looking at the.
A sea spider?
at the yeah.
Is
Well,
it feeding on something?
I cannot figure it out.
Are we zoomed in, Max?
No, but I.
Can you zoom in on the
I will.
proboscis? It's feeding.
Yeah, I'm not entirely sure. Look at that leg back there. I
Or
don't know
is
what
it?
Well, I don't know what to do.
Oh my god. The funny thing is that on
We're not
land,
little.
I really do not like spiders that much. It has gotten better, but
Well, this is
this
a
isn't
kilometer off from you, so you're a bit of a chicken here.
Really, yeah, I know.
Now you
no,
like
but
it
no,
more.
I really love this.
Come on. Yes, of
I
course,
really
me
love
too.
this.
From the distance.
It, actually,
I'm not. I don't mind spiders, therefore.
no.
Do these have fangs?
They have a what it's called a proboscis. So what you see the main the core body
Yeah.
In dark
And-
red here.
in dark red, and then you have the legs, and in front between the legs there's an elongated sort of oval shaped structure, and that's the proboscis, and it would use it to suck.
Yeah. Suck food in.
Exactly.
So-
And we wonder
I,
whether it's.
well, I know too little. We're losing it here. I know.
Oh, now we're
Up,
Yeah.
till up.
there. We are.
Bob's moving.
I also know quite little about
So-
them.
So I don't know if their mouth parts extend in both directions. That confuses me.
Exactly, and are these? This belongs.
yeah.
It belongs to this,
It's
the
a sea
animal,
spider,
right?
so it's picking something up.
It's
They
just.
call it, so you don't need to stop creating facing the
This is incredibly cool. We really
We have a bit of a sea spider.
we do. We have something going on here.
8-frame bump stop. Docking head bump
Wow.
stops.
Well, some
Look what it
But
does.
it's cleaning itself.
its legs.
Oh,
Oh my god.
the spider's telling us something. Have you cleaned your cabin, Jenny?
Well, it's cleaning. Look how gently.
Yeah. We encountered other sea spiders where the male has all the little ones on the back, but I'm pretty sure this group doesn't do that.
No,
So not all sea spiders
no.
have this paternal care.
I just want to get everything in the picture, you know?
Nope,
No.
you just sit and watch.
Yes.
Do we know why they're coloured? Because there's no light down here.
So, I don't know why in particular, but the red color, we only see this. So, red is the first
Spectrum.
color of the
Spectrum.
spectrum that disappears with depth.
Yeah.
So red isn't actually really a color in the deep sea if you
Yeah.
put it that way. Which is why lots of animals have like reddish color, like the anemones.
Yeah.
And all the shrimps, for example, because it really disappears in the deep.
Camouflage.
Yes, it is.
Makes sense.
There we go. We're back to slow sea spider TV here.
Mm-hmm.
Should we do a media change?
Oh yeah, definitely. Ooh.
Sorry, I'm so distracted. Yes, please.
Thank you, Paddy, for keeping track on our
was our job.
tasks here.
Okay, 3, 2, 1, stop.
Yo. Actually, it,
Dry lab ROV.
Ich verstehe nicht.
Dry lab ROV.
Right lab.
Yeah, media change.
Yeah, we're changing.
again, I could stay here forever.
Okay, media start in 3, 2, 1, start.
Yo. So we're at 833 meters depth. We're looking at ascidians and anemones next to a pretty large sea spider. We have some clams, soft corals, polychaete tubes, encrusting sponges, bryozoans, really diverse habitats, really diverse rocky sea bottom, incredibly beautiful and rich. This was lovely.
Yeah, thanks a lot.
Thank
for
you.
Happy days.
letting us
Happy
have the time. Yeah.
days.
Look at that. What?
Turn a bit up. Happy days for one last shot. If you just put a bit up.
I just want
Yes.
to see spider on it as well.
Ah, awesome.
Moving on.
Yes, please.
Sad.
Yeah, a
Yeah,
little bit.
it is a bit
I'm
sad.
kind of waiting for the dry lab to tell us to put the sexton out.
Yeah,
but
same.
I don't know what's up with the dry
I
lab.
don't know. We can put the lasers back on, please. Cheers. We have Rosalie Day again.
It's nice.
Soft coral garden, or like abundant soft coral. Skulle jag bara få So, what time is it? 10:30. Okay, no. I'm confused. 9:20.
This is slightly confusing with this time, and also with the keyboards actually for typing, because they're English and German keyboards and they're different settings.
Yeah.
So someone reading these protocols have a lot of spelling mistakes.
Yeah, for sure. Lots of Y's for Z's and the other way
Yes.
around. And some question marks for the. Oh,
Mhm.
what are we logging?
Starfish.
Really? Oh
Sea
yeah,
star.
there it is.
Yeah.
Oh, I was confused because we have UTC here on that clock, and then I have another time on my phone, and then I have a different time again on my watch. So it's all a mess. Oh, there's another sea star.
And lots of soft corals.
Lots of soft corals. Uppermost. Sponges. Very, very pretty. And I think lots of the brownish little balls are Carniellas? Maybe not.
These ones are Ascidians.
Are they all Ascidians?
Yeah, look at it. You see the two openings and some of the ones that come into more detail.
I was talking about the orange little.
Oh, so, yeah, no, these are sponges.
This is a Carniella.
Yes.
The golf ball sponge. Beautiful. Really beautiful.
Right, Jorvie, we resume the move, please.
Rosalida ahead, I think. And these vase-shaped sponges, if I'm not mistaken, amongst lots of soft corals on a rather rocky or like pebbly sedimented seafloor. We still got lots of anemones. pretty.
And all these worms.
And all these worms, you're right. Dense.
Dense aggregation of polychaetes.
Yes.
So let's
Quite.
look at that sponge.
Yes.
Just ahead of us.
Yes. Wow. Oh no.
Look, yeah, it's over.
Yeah.
Can't
Yeah.
do anything about that.
No.
Lights.
It's alright. It's alright. You know what? I feel there's quite few fish actually. Or have you seen lots?
And few cucumbers.
Yeah. Yeah. I would expect more fish. More. There's one.
I'll show you one. Come on.
Okay, thank you.
Here you go.
Thank you.
You can
Now-
walk now. Fish now.
Thank you. Thank you. That's perfect. It was about time, you know. I logged Grenadier to be precise.
Thank you.
These sponges grow in such
Weird
weird
shapes.
shapes. I mean, it's really nice. Wow. Wow. Now it is really a soft coral garden. So rich. What is that? It's
Can
a
we s-
feeding sea star.
Okay, can we slow down, please? There it is. But I don't mind slowing down.
We haven't had such a large specimen, maybe? Or have we?
No, I haven't.
No, me neither. So we're having a large specimen of a sea star ahead.
To be truthful, I was more interested in the borers than in the sea star.
That's all right.
Yeah, but it's quite hard to see if it's really a borer or if it's some giving like a dark reflection, like a shadow.
Yeah, yeah.
And something has a bit of an overhang, like a little rock. It's quite dark underneath, so it's.
We can continue. We don't.
Yes,
It's
we can
fine. It's
continue.
fine. It's fine.
Fine,
Okay.
thank you.
But it's nice to see.
I agree.
Estrella. It's quite big. 40 centimeters or something. So we started the dive at 1,089 meters approximately, and now we are at 808 meters. So we've climbed quite a bit.
Yep.
Wait,
Was it jelly?
I think so.
And we're almost finishing our transect here.
Yeah, we are. And then we've got another one ahead. Was that another jelly?
And a lollipop ahead.
Uh-huh. where you're spotting?
Another softball.
You are really looking for the sea cucumbers, aren't you?
Maybe it's
Maybe.
yes.
Yeah.
It's another sea star.
Ah. How far is that blue transit there?
Do what?
The distance between the EOL and the SOL.
meters.
470?
Meters.
Okay.
Because we are at the end now of our transect.
Mm-hmm.
Pretty much, yeah.
And what is your recommendation then to take the rope up
No.
and have a flyover?
Yeah, fly
Yeah.
across.
Yeah.
Yeah.
We can do that. Do you have an approximate estimate of how long it will take us to fly across?
Maybe 40 odd minutes. So.
40 minutes?
Yeah, a long way.
Okay. Shall we
Can we have
check in with the dry lab maybe?
maybe let's have one last zoom here while we're here.
We can do that.
Maybe we can
We can always do
Okay.
that.
yeah, okay. Maybe we can just
Nice bit here. Nice bit here.
Yeah.
let me know whenever you're ready, because we're at the end of our transect here. We'll continue our fourth and last transect on Schulz for today in around 40 minutes after we finish this one, but we're not there yet. We want to zoom in one last time. Oh,
Hello.
yes, the master of knowledge. At the end, so we're just saying goodbye to T3, and we'll have another close-up.
Okay, it's all yours.
Thank you.
Thank you. There's the Solasta. Hey.
that's really-
Hey.
Just settle it down.
I was very busy
Thank
with
you very much.
logging, so I didn't even see it. Lollipop and Solasta and lots of things happening.
All yours.
Thanks. Thank you.
Wow. Amazing.
I'll turn a bit to the left.
Oh, to the left.
Yeah, sorry. We'll come back to them. They're not that fast.
Okay. What is this rounded? I will actually have to log round sponge because I cannot identify it. Canela. These are pretty soft corals. Hello. You are scouting for amphipods again. I
I
can
am
see
scouting
it. I can
for
s-
any forms here, not only for amphipods. Come on.
Which?
Where's the sea star? Sorry.
On our right side.
Okay.
You'll get it as a goodbye of this one, okay?
Four.
I thought you were well, right in the middle.
Thank
It's
you.
just it's just hiding.
I haven't spotted it yet.
No? No?
Oh,
No?
what?
Maybe I'm wrong.
I don't see any whelk. I see a
Maybe it's in the city.
You
and
want
I
us
thought
just
it's a
to
siphon.
continue blue water to start at T4.
Yeah. It's
Yeah.
one stretch.
What was it, 470 meters?
477 meters,
Yeah,
yeah.
450 meters.
Because we still have, I mean, it's
Beautiful. I think
Ships are moving.
that is
Yeah.
in Colonial City.
2 more hours or a bit less.
What do you think?
So you'll just have
Yeah.
start.
I first thought it's a gastropod, but I think you're right. You see all this.
So that transit will only take us.
I think it's a Colonial City and
Let's have a look. Worms. Look at that. Look at that one. Wow. No, that's another battle star. Yeah.
we could continue the climb for a bit longer. Just it's a nice slope.
Whatever. It's all good. Whatever you decide.
Because the new transit will take us for too long.
It is beautiful to just continue up.
Wait.
Let's do that.
Hmm?
It'll nearly be the same line.
Yeah.
So if I go here, we're on that course anyway.
Great. Let's do that.
So all
Just
you,
do-
all you have to do is
I'm just zooming into this sponge.
just lengthen this, and you're actually working.
It's better.
Okay, so
Wow.
Let's do that.
okay. And we still come off bottom at 10.
Please
you can
note
see.
the little critter sitting inside.
Well, if it's just 40 meters, I
Yes.
don't know.
Can you turn off the lasers, please, Rob?
Thank
Or,
you.
thank you. Wow, this is so cool. So we have an Asconema foliatum, if I'm not mistaken, to the left, with some really pretty ascidians to its lower left, and then we have this vase-shaped, what I believe is the sponge of the Rosselidae, to the right, and we can look right inside it, which is really beautiful. We have tiny anemones and larger anemones. Lots of life. Really pretty. Do you want to add anything about little critters there, Anna?
No, I'm trying to get them in focus.
Yeah.
Okay, bridge all stop.
You see, well, you
But
see
maybe
this
you
little
need to-
critters inside the sponge
Yeah.
that we're looking at now? Well, they are amphipods.
I have to look. I'm still waiting for my little sea star zoom there. I have to say, I'm not quite satisfied yet. Where has it gone? I knew we should have zoomed on it first. Is it really gone? Excuse me? Excuse me? This is not true. No, look at this, I found you. Or I haven't.
No, that's not it.
Oh! Really?
That's not.
Oh my god. It disappeared. Well, I really wanted to show you. Ah, no, there it is. It was just a bit far off, you know? You see? Lurking in the back.
A little bit to the left.
Yeah. It's very far away though. Yeah, that's a Solasta. Pretty sure.
It's also having this one arm up.
Yes,
Maybe that's what they do.
you know, sensing, looking, because they have eyes in all of their arm tips.
Well,
Each arm tip is one eye.
wouldn't call it an eye, but
Okay,
it's
or
a
seven.
sensory.
And a
cells
celum.
is-
Okay.
yeah, I agree, but I didn't know that they only hold up one.
Okay. I think We have.
That's just been changed.
We
Sandra,
have-
do you want to look at anything in
Do you
particular?
want? Shall we?
Get anything for you? No.
No, but do you want to look at anything while we're here in this most stable position?
I'm following you from dry land.
Okay.
Oh,
All the time.
you know you don't have to. It's just
This is very pretty.
Jenny and I are just so focused on the small things that
We never really make.
we make a lot of.
I am actually
meters.
surprised we made it to the end of T3.
Me too.
Excuse me. Like after how many hours?
We'll just continue.
yeah.
Continue
Okay.
northward and
Yeah.
oh, and there's the tiny little stars.
I mean,
They are so numerous.
we only used 4.5 hours for a couple of 100 meters. So.
Well, there's a lot to see, guys, right?
Well, that's why. And as soon as you stop, you see more and more. And it's also.
That's what we're here for.
Mm.
We're not here to get some mileage and cover as much distance as somebody thinks we should. We're here to see life on the seabed. Yep.
Amphibians.
What's this slope down here and up here?
You tell me.
No, we're making. Well, you know how some people always focus on their favorite groups. So I just have to point out all these amphipods.
Mm.
And there are two in the. There are actually three in the picture now. Right, like right on the bottom line in the middle. You can see it's crawling upwards. It's in the middle here. And you've got one on the left corner, to the right of the white anemone. You've got one here on the sandy patch, and the longer you look, the more you'll see them.
Exactly.
But I, of course, appreciate that the first thing the eye is drawn to is this pink anemone.
Yes.
Definitely. I'm
Hmm?
good.
It's got a long leg.
Beautiful. I think there's this encrusting sponge again that we sampled earlier and want to figure out whether it is whether it is something new or wow, look at this soft coral.
You mean this one?
Yeah.
Look, we have the little anemone growing in between.
It's all bananas.
Wow. And some carnivorous sponges too.
Carnival pilots over here. I take it.
Are we having snacks?
No, it's having meetings. Just having meetings. But you want some?
I don't
You want to come into the meeting?
hiding there in the back.
Chocolate for it.
You're omnivores, you eat everything.
It's a really snackish dive here. Oh,
Thank you.
How are we doing,
thank you.
ladies?
Yeah, we are.
We're focusing along back and forth, thanks a lot. And we will soon say bye.
Yes.
Okay.
Okay. Yeah, I think we got it.
Mm.
You don't think so?
Mm-hmm.
Ready to go?
Mhm.
Yes.
Okay, bridge ROV. Can you resume the move, please?
Sorry, so please ignore what we had said about the blue transit to another site. There was a change of plans. We got told now that we were allowed to continue on this slope.
What would you
can have the lasers back on.
Sorry?
I'm just getting the transect. I'm just going.
Keep it on straight on, yeah.
Okay.
Thank you.
Yeah. Yeah.
Is this a no stopping zone, is it?
No.
So
It's like the light, basically.
for those of you who would like to know, we have a current water temperature of minus 0.1 degrees Celsius. And a depth of 793 meters. Oi!
Can we slow down,
Oh
please?
wow! And we're look at that sea star! Oh, and we're back to an amazing, beautiful rock. Ah, this is just great. Now, excuse me, but I need to zoom on that sea star.
No, you'll get a better one.
Oh. Porania? I think Porania. Wow. Can we start?
Dry lab? Are you sure you don't want the sexton out?
Hmm.
Well. Oh well.
This is perfect, Rob. Thank you.
Rob said, try that. Please push up the thing again.
Okay.
I'm not sure what happened, but all these anemones pulled in their tentacles all at once. So all this, I don't know, pinkish blobs.
Okay, it really does that.
They really want us to focus on the sea star for once, okay?
No.
Please.
Look at it.
Look how beautiful it is. It looks so
Sea
soft.
It's a bit like a cartoon.
Sorry?
Looks like a cartoon figure.
Yes. I think Porania. Pterasta, Poraniomorpha. Not Pterasta, maybe. I will log it as Poraniomorpha. Yeah, nice. And lasers off, please.
Off.
Thank you.
It is a cutie.
It is a cutie. It really is. Soft little, chunky, chubby.
Andrea has a good taste, our off-pilot chili grace.
Okay, I'm happy. I'm happy with the sea star footage. encrusting yellow sponge.
Okay, let's continue.
Yeah, we're good. Now there's a little plume here.
Okay, so back.
Bye bye. Ah, carnivore sponges. These
We
rocks
can
are
continue.
amazing.
Fine, thank you.
No, carry on.
We can carry on with lasers on, please.
On.
Thank you. Oh, look, there's a round sponge on top of another sponge.
I think it's an enemy.
Really?
I'm not
Okay,
sure.
Bridge,
I
back
thought
on the
so.
move again.
No.
Nope.
I disagree.
Oh, let's go to opening one.
Nah, I disagree.
That's fine.
Asconema foliatum. Another lollipop sponge. Stylocordyla borealis. I learned today. This is. It just continues. The beautiful habitat never stops. It's just incredible. This entire slope has been so beautiful.
I completely agree.
Wow. Lots of these yellow encrusting sponges. If you just focus on the color yellow, you see them everywhere. It's really nice. You see, it's the same. I think it's a sponge.
This one, that
Yeah.
one, yes.
Yeah.
Yes, that
Yeah.
one for sure.
It's really pretty.
But I think we want this pinkish blob on top.
No,
Maybe let's
no,
look at
no,
something
no.
else. Because
Maybe Paco has texted rather. Yes, logging the Stylocordyla borealis, the Rosselidae, and what's that?
I can't see.
I'm seeing something toward the right.
Yeah. Oh, it's our Tubeworm.
What?
No.
Can we stop, please?
Can. That's one there.
No, that's
This is something
that's
new.
That's a stalked.
But this is new.
Yes.
Let's please.
I forgot the name.
Umbellula.
Umbellula.
Is it? Is it Umbellula?
Just have a double check.
This is great. This
Yes.
is new.
Because I was waiting for that.
This is great.
It's a sea pen.
There are more coming.
Yes.
The bridge also.
Well, there should be quite a few coming. And
Okay, this is exciting.
and it's,
I'm very sure it's Umbellula.
you remember Paco talking a lot about Linné, whose birthday it is? He described that species.
Bottle of water.
if it is that one. Umbellula
It's
and Crinus?
Umbellula and Crinus.
I think
Yes.
so. That's Linné. Linné 1758 or 50.
58.
Anyway,
fusion.
I'm not sure. 50 something, but definitely described by Linné. And they have been observed on this seamount before.
Yes, and they are amazing. They are stalked anemones. Can we call it that? Is it? I haven't really. I've never seen them before.
It's a sea pen.
Wait. I'm excited.
Pretty sure it's also one of the indicator species of the vulnerable marine ecosystems.
Wow. Right, sea pen.
I'm quite happy to fly on. I strongly predict we see a lot more.
But I have a question. Are we heading? If you see on our camera now, I can see that there are several umbellulas to the starboard side. Is that our heading? Not really.
Let's just keep
heading
here on the
is-
right side.
The
Okay.
heading is but lateral. See, we're heading straight that way, but I've just come over to the
Okay.
starboard side.
Can we
Can we
just-
just stay here
Lateral.
on this side, please?
Is that okay?
Yeah.
Thank
Yeah.
you.
So you want to fly up towards it?
A little bit, yes.
Just that we would just want to see if the field gets more dense. Since it's the very first specimen.
I think so.
Now there's the next one coming in.
Yeah. I really need to just look up something about this.
Okay, Bridge. Let's resume the
This
move.
is it's incredible.
Yeah. Sea pen.
Yeah, it's a sea pen.
It's
Wow.
Umbellula and Crinus.
Wow. Yeah, I wanted to look it up. Linnaeus 1758.
Told you.
Yes, amazing. Amazing. So excuse me that I said stalked anemone, that was completely wrong. It is,
Well, it had the color like
Yeah.
our sea spider.
yeah.
That's also a bit
Yeah.
confusing.
But look, they're not.
Hmm?
They're not. We're only two so far.
So we're not really flying up the hill here at the moment. We're just flying along it.
Yeah.
Yeah.
We.
We'll return to our track. We just wanted
Yeah,
to see if
sorry.
there's
Okay.
like
It was just like.
to see if there.
All right, fair enough.
Sorry, to have a peek over
Yeah, yeah.
the edge.
No worries. No worries. You're turning at a quarter of the road.
There has been a little slide here, a little landslide.
Mm.
A little, well,
Yeah.
you know,
Right.
a landslide, a sediment slide.
Mm.
Yeah, back up again. So yeah.
Oh yeah.
So that was quite cool to see Umbellula and Crinus on Linnaeus' birthday,
Yes.
described by Linnaeus. Really nice. Ooh, another bit of sea star action. Hi Paco.
Hey. I've seen that you were super lucky
Sorry.
as well.
Lucky.
With the
Yeah, to
rock.
see with the sea lilies and
Yes.
and the
Lucky,
giant
yeah.
sea spider.
We are very lucky woman today.
We just found the first Umbellula.
We go to point
in
2 Bridge.
Crinus.
Yeah, the
first
stock
two,
crinoids.
yes.
Yeah,
It's
yeah, I
a-
saw.
it's
no, it's not a.
Sea pen.
It's
It's a
sea
sea
lily.
pen.
It's a sea lily.
Thank you.
A sea pen.
Sea
Yeah.
pen.
And it's
Bye.
Yeah, I saw.
Yeah.
And Anna just mentioned that it was
guys should
described
go
by
on that yourselves.
Linnaeus, so
Ah,
it's really nice.
No, no, it's not worry.
It should be there then.
but it also
Yeah, but
shows
Mac
that
is
he
connected to
described
this.
this
It's for you
deep
guys to
water
pull across.
species
Okay.
so long
Let me
ago.
know if it's, if you're
It's not only
if
what
you're not
was
seeing
found.
it.
Well,
Yeah.
it's what happy to be
yeah,
the last
absolutely.
couple of
They had
days.
a huge interest.
He has
But is a network there or is it just not pulled across?
described a lot of
I'll double check.
Yeah, no bother.
marine species as well. Nice. Thank you.
I'm happy to stop again if you
If one of you,
I would like to get myself a warm tea. I'm just cold. I'll
Yeah.
be back in a...
yeah, sure.
But you can do some logging if you like. We
As you
haven't
wish,
really-
we can
haven't-
exchange if you,
let's
because you've
swap.
been in that seat for a while, and I know that seat.
Thank you. Yeah. Is that yours?
Yes.
Ah, there we go. All right. Cheers. So
must be also. These are big, very big.
Yeah.
So it's a lot of people also ask me how old these sponges are, and it's very difficult to say because there hasn't been so many experiments or research done in this field. We do know that Arctic sponges probably grow very slowly. And the few measurements suggest that sponges that were maybe 20, 30 centimeters large were more than 200 years old. So this gives us an idea of how they can be.
The woolly sponge. Was
Oh
that
yeah,
it?
yeah.
The woolly sponge.
Yeah.
I haven't
It
logged
is.
that. You can log it.
Yeah. But here I feel I will need to log like every sponge we've seen so far because there's so many of them.
Yeah.
It's wow.
That's beautiful.
Yeah.
And also really nice and gentle climb here of our pilots. They are very patient with us.
Yeah.
You know, we fly for 10 meters and we're like, oh, stop. And then we, there's always something.
Our pilot currently is Rob and co-pilot Paddy. And they are very skilled. So if you ever spot another Lysodendorix, the pompon sponge, I'm interested to collect another one, maybe in one of the bio boxes because we still have a lot of space.
This is Aspenema foliatum?
It is.
Okay, then I have locked it correct. I was a bit
Yeah.
uncertain, but
Yeah, it is.
thank you. We can very slowly just move forward. Thank you.
Have you collected something for our photographer, Solvin?
We have.
You have?
You know, in the beginning when I was interacting on your sampling event about the sea star,
Yeah.
that was actually a request of our photographer Solvin Sanke, because the Hymenoster sea stars are just very pretty and they're very beautiful to photograph,
Oh, okay.
and we need it for our collection as well.
Yep.
So there were two
Great.
reasons to collect one, and since we saw
that's-
several.
I just saw him. That's why he's so excited. I didn't know
Yes.
that the sea star was for him to take a picture of. So
Sea star.
yeah.
Wow. It's really cool. Wow. And you just say it again. It's how did you put it so nicely? With the
Yeah. I just said every wall is a different painting. Yeah. It's
wow.
it's pretty amazing how colorful the Arctic deep sea can be.
That's a Poraniomorpha.
Al's right sea star.
You have a chubby one
Yeah, I see it.
and another one.
Is that the one we sampled on the previous dive? I think we actually gave one to Solvin, but I don't know if that's if it's that species. So here sponges are essentially of the family Roselidae, those vase-shaped white sponges. They are glass sponges. They live essentially in the deep sea. Glass sponges you will never see in shallow waters. This is really what they like, the deep sea, cold. Right now it's we are plus temperature now.
Really?
0.05. Yes, we are at zero, pretty much,
Wow.
but a little bit plus. But we're getting to the top, so I think we have maybe changed water mass as well.
Yeah, I mean we are at 730 meters, so we
Yeah.
have a different water mass than what we had when we started the dive. Oh my god, this anemone coverage is just insane.
Yeah. No. So this is turning into anemone country.
An enemy country.
It's okay. It's okay.
I would say
I-
an enemy alley. An enemy, whatever. It's so beautiful. What is the yellow?
It's a sponge.
Yeah, but do you know?
So we think it's the genus Aplyzilla. It's a sponge that doesn't have a skeleton of spicules, and they are very rare in the deep sea. They prefer super abundant in tropical waters, but in the deep sea, very rare. So, to my knowledge, that's the only sponge without a skeleton that we've seen in the Arctic so far. But it's pretty much everywhere. So,
this is beautiful. Sea star.
yeah, because it adds a tinge of yellow.
This is a hormatid.
Turn your head as you come onto that ridge.
Hormatid.
How do you write that? H-O?
Yeah. Hormatid. Yeah. An enemy.
Anemone.
More northerly. But on the ridge if you can. It's a lateral portrait.
Beautiful. We have seen lots of beautiful
Is that
habitats.
another one?
I think so, yeah. We've seen so many beautiful habitats, but this is special.
Yeah.
Come up on the ridge.
This
Yeah.
an enemy garden, like, wow.
Yeah, because it looks very tropical to us, although we are in the Arctic, in the deep
Exactly.
sea, in the middle of Norwegian Sea and Greenland Sea. I hope people watching this realize that this is probably against the idea that they had of the Arctic bottom.
So this is how
Ocean.
the deep sea actually
Yeah.
looks when you have hard bottom, some current. You are at 700 till 1,000 meters. We've had this band now
Mhm.
of really like dense anemones, soft coral, sponges, all these colors and critters
And no bottom fishing, because
No.
if you would fish there, you would absolutely destroy everything.
yes.
So this is untouched, pristine sponge ground.
Mm-mm.
So we are beyond our transect right
We
now.
are.
Oh, I get it.
We
okay.
agreed that we would continue on T3
Yeah.
because it's just so lovely. It's
Yeah.
so diverse,
Yeah.
and why stop this amazing
Yeah. Yeah.
exploratory transect, right?
But we are still going up. Yes, we are. Okay.
We're climbing,
Yeah. Yeah.
climbing. I bet it's quite difficult to maneuver here because it's a quite steep slope. You have lots of three-dimensional habitats, so which you don't want to destroy,
Yeah.
obviously. Like
It's a portrait and
Yeah.
then
you need
lateral
to
starboard,
constantly maneuver with the depth, and it's very challenging.
That's a perfect opportunity for me to explain a little bit why we call the big animals here ecosystem engineers.
Oh yeah.
So all the big animals like sponges, soft corals, anemones, anything you can basically see on the picture, are ecosystem engineers. They are building the habitat by being large and creating lots of different niches for other organisms, much smaller, that we often see when we stop and zoom and take our time. And these ecosystem engineers are truly important. These are the ones you need to preserve if you want to keep the whole biodiversity and the whole habitat. And that's why sponges and sea anemones, oh look at that, man, it's getting denser and denser.
Can you make a note that this is the most... Sorry
No.
to interrupt.
No, I think I was finishing with my point. I just got distracted with the density of anemones.
It's amazing.
amazing. Look at these colors.
Wow. Can we go a bit slower, please? Just a little bit slower. Thank you.
Mopshak, can you turn off the laser?
And maybe we can settle and stabilize a little bit to appreciate the
yeah,
view.
We won't be able to land, but we can try
No,
and
not
stabilise it.
land, but just
Yeah,
that would be great. Just stop.
start it up there.
Wow.
Is that telling
It's becoming a habit now and stabilized.
Yeah.
Once it's stabilized, our off pilots can have a break. We have a play with the zoom, and then they go to the snack box.
Uh-huh.
So you tell us when.
You problem.
Hmm?
There's a tree there from
So for us, it's 10:30. We are 1 hour behind. 10:30 p.m. We are 1 hour behind Stockholm or Paris time,
Dublin.
Dublin time.
Can we turn the RV a little bit to the looking to the left? Yes.
And because we are beyond the polar circle, we are 73 degrees north. We are also enjoying the midnight sun every evening. So the sun never completely sets here. And the temperature out is around 5-6 degrees. But in the water, it's currently 0.08. So it is much colder. And when we sample organisms at these depths and bring them back on deck, we always try to put them in very, very cold, icy water to try to preserve them as much as possible, especially if we want to make pictures and we want to make observations, or we just want to study them.
Okay.
Are
You tell us when we're stable.
Okay, bridge, all stop. Oh.
So we're going to wait a little bit for the sediments to clear out.
Oh, but this is quick.
Oh, yeah.
There's so much current.
Yeah,
Wow.
Sure.
it's like sponges in the mist. Oh, yeah. Wow. Lots of current up there.
That's why these anemones are so happy.
Yeah. When there's current, there's food for all these animals that feed in the water column, like the anemones, the sponges. Oh, wow.
Wow, that was,
Yeah.
that's awesome.
That's incredible.
Are we?
I'm sorry, I'm taking
are we
another picture
stable?
because
Yes. May I go ahead with my Zoom?
Yeah.
Thank you.
this one? Or is this, no, something else?
I dare you.
Hey, Jane, how are you?
Dare you, beat you.
You think I'm
Wow.
Okay.
Your nose, you bloody.
You match your twin, don't you?
So the big white cabbage looking animal in the background is a sponge. It's a glass sponge.
Look at this, Jenny.
Oh,
Jenny,
yeah, look.
Jenny.
No! That's
That's a nudibranch or
it's a sea angel.
It's an angel,
Yeah.
exactly.
Oh, it's Cleona. What is it called?
Just
what
Just
what
enter
type? It's a mollusk.
Yes, it's a mollusk. It's a mollusk. It's Cleona limacina. I just have to quickly. I haven't used this.
but they're
species
called sea
before.
angels. That's sure.
Yeah, sorry. This is just my geek.
I know
geeking
that. I know, but it's also one of your favorite.
it. It is Cleona limacina. Yes.
Ah, that's it.
Just enter sea angel. That's fine.
In German.
I just put my finger on it.
In German, it's Meeresenge. That's
Mhm.
also really beautiful.
Paco, you refuse to enter that, don't you? Meeresenge? Oh, what is it?
No.
The largest snake.
No,
I'm
looking at
just
this part.
I'm just mesmerized by
Yeah,
it.
me
So
too.
I'm dreaming about it. I'm still dreaming about it.
I almost missed it. Thank you. I was caught up in.
You're snacking back there.
I'm not snacking.
You're still chewing, don't lie.
Stick out your tongue.
Oh my god, this. Yeah,
Okay,
let's just
sorry. I'm
stay.
gonna take another
Is
picture.
that good? Yeah.
Let's
I
just.
want to get this overlit.
Hey, how about we do our light thing again?
No, I want to get this out of the way. It's too reflective.
You know, like.
Yeah, one-sided lighting.
One-
And Anina said, no, wait until she's finished.
That's correct.
So we shall do that once we're told.
Okay.
Yeah? Camera
Yeah.
lights action?
Yes,
Yeah.
please.
Okay, let's make this happen, right?
Oh, very good.
I already like it.
Yes.
Me too. Shall we just move a little bit further to the right?
Yes.
So we don't waste so much screen with blackness?
Or below. Don't you have more?
Yes, but no. Too much to the front.
Too much off.
Yeah.
Too much
How
off.
we
Too much
overdid it.
reflection to the front.
That's
That's
Yeah.
nice. Yes.
Still move a little.
nice, nice.
Hmm? How about this?
Yeah.
If we zoom in a bit, we get less, just a tad, like
You will get less reflective.
yes,
I see what you mean.
yes.
Yes. We
But
can
then
do-
again, the effect of the
Oh, of the
this-
gilded one.
yeah, yeah.
Mm,
But I love
This
this.
is, come on,
I love
this
this.
is,
the light.
Thank
this
you.
is
Yeah.
very
It's
nice.
been a long day. It's been a long day.
Hmm?
I know. I've been here too.
Okay.
Ah, I love this.
Me
Just
too. Okay.
Oh yeah. Left eye's winking at us.
wow.
She didn't see his hair, but that's quite cute. Oh my.
Okay.
Big floppy nose.
We have enough.
Yeah.
No? Just looking.
Is that his ears or his cheeks?
Could you zoom on that after
On
when
which?
you're on that sponge? I'm
This one?
not quite sure.
Oh, yeah.
Yeah, this one.
There's the Arabic.
I'm not quite sure what this is. Ah, yeah, okay. It's the Helicondria. Yes,
Yeah, you take the other one.
good.
this looks unreal. It's
Well, it's 700 meters below us.
just, you know what
Mhm.
I really want to do? Keep a zoom on the anemones and then switch off the lights to see if they glow.
They won't glow. I
What do
check.
you know?
Oh,
Hey,
Remember that, right?
I see.
Yes.
Going in my brain.
but I think it's a good idea, Jenny.
We don't have to do it.
No, but I.
But let's do a little zooming first and then have an
Hey,
overview
hey,
with
I
the,
charge
Yeah,
you.
yeah, yeah.
with the lights off. Hmm?
Transparent silver to give out a yellow light.
No, the ones that's wrong. The one they're pink is in.
Thank you for focusing on the geodias, Ananina. Two beautiful geodias here.
Anyway.
It's nice. I know it's not what you're looking for, but
It's exactly what she aimed at.
You feel your pain.
What are you looking for?
Me?
Yes.
Oh, I don't know. There's always something.
Hey,
Okay.
let's zoom in on sediment. We haven't done that before. No. But you know what?
I think it is quite interesting how the sediment is made
but
up.
I don't see any amphipods.
it is getting quite late.
Well, not sure. I think I have some prejudice that I'm only looking for amphipods. So we'll go back camera main position and we should. Can we here turn off the lights, please?
So we're going to turn off the lights entirely. And this is.
No.
No.
Are you sure?
Yeah.
Well, otherwise we would see.
So all of these dots are just
No.
No.
nothing.
You can turn them back on again. Thank you very much.
Well, we had a taste of what the deep sea looks like
Yeah.
if we're not there.
The real experience.
Hmm.
Okay, dudes, are we up and away or are we waiting?
We can continue?
You're the dude.
Yeah. Okay.
Yeah.
Okay, Bridge.
Then let's go.
Get on the move again. Say, resume the move.
I say in the middle of that ridge we can't.
Sorry?
I say in the middle of it. That's it. Top man up.
That was beautiful.
That's just what I stood in. Behind you.
What is? Is something more?
Yeah,
Is
there's
that
a
squid?
grenadier.
Ah,
I
okay.
think. Yeah.
What is
Ah,
coming
yeah.
up?
Okay.
It's a fish.
A grenadier
Yes,
fish.
yes.
Yeah. It's very chilled. I mean, it probably has never seen a robot in its life, but it's very chilled.
But it's still alive.
And it's funny because the anemone stop. They're gone. There's so many before. What is going on?
They need a very specific.
Yeah.
They need a certain current speed in order to feed and
I mean,
grow large.
yeah. That's something that really strikes me on any seamount that we've been. There's these abrupt
Yeah.
changes in communities, like 1 meter, 2 meter beyond,
Yeah.
and
Can we have the lasers back on? But that is quite a big.
that's a big
Fine.
one. That's a big one.
Thanks.
So we have about 20 more minutes of this beautiful seafloor,
Yeah.
right?
Mm-hmm. Yep. But I would like to collect the geodia before we end the dive. At some point, if you see a geodia that is nicely covered, especially I'm looking for the animals that grow on top of them. Moving or not?
Movies? What do you mean?
Moving amphipods or not? Not moving, encrusting.
Oh, I thought you meant like an encrusting sponge.
Yeah. Of course, I'm mostly interested in the encrusting sponges, but it can be other things.
Okay. More?
Of course.
Go on then.
I'm just thinking about our coffee mug, but there's nowhere here to sit.
Oh, there's places where there's a bit less of. Wow, look at this! It goes on and on. This is huge.
Wow. This transect is so beautiful.
Mm-hmm.
I am very happy we took the decision to just continue the climb.
Yeah.
Because
Yeah, me too.
this footage is brilliant. As soon as there's like a little rocky outcrop, the anemones are back.
Well, you were wondering where they are.
Which ones?
Well, yeah, but
The anemones.
those 152 on the screen. You just said that there are no anemones anymore.
Ah, yeah. There they are. Yes. Yeah, but a few meters behind they were gone.
Yes, I know.
Mm.
I think they're a little bit to our right. See it on the other. Oh no, the big ones are back.
Mm.
There's the Actinus dolida, I think.
The anemone?
Yeah, I think it's Actinus dolida. Or Homatida. The thing is, sometimes I have difficulties to distinguish them because obviously
I mean,
I'm not,
no, but also obviously it's difficult to see. We're a bit
but
far
I
away.
know that there are these two families that are both abundant in the deep sea.
Hmm?
And I am quite sure that we have both families here present, several species of these families.
Oh, there are a few things missing, like our sea cucumbers.
Mm-hmm.
I'm surprised.
Maybe they don't
And
like
also sea
spiculmats.
sponge. Yeah, that's an option. That's actually, I hadn't thought about that.
It's maybe
That's-
very
that's a
uncomfortable to walk on
Yeah.
spicules.
Yeah, and to eat it. I mean,
Mhm.
that's
Yeah,
what they do.
Yeah, exactly. Yeah, they
They
need to.
filter through that makes
sand?
complete sense. I hadn't
Yeah.
thought that through. Yeah. No, you're right.
Ooh, that was a station. So in one of the previous dives, Julio tried to sample this kind of
Yeah.
thing, thinking it was a sponge. It was 100% spiculomat.
Wow.
And yeah, it's very thick, and sometimes it takes shapes as if there's sponges hiding under it,
Alright,
but,
but slow to point one.
but it's not. So we've
It-
tried that.
Dear pilots, if you see a place to land, let us know. Because
Okay.
we still have the coffee mug.
All right, yeah.
Cheers.
Thank you.
What you have to get sediment, sand.
Sediment, and if we can collect
No.
one
No. Focus
sponge.
on, focus on the mug.
Turn your head to that way.
Oh, there was a tumbling soft car.
Would you
Mhm.
like to try that?
Go ahead.
You can wrap up the live stream.
Okay, thank you.
Thank you. Goodbye, listeners.
Well,
Can we take the sextant then now?
Yes, you can do that.
Okay.
thank you for following us today on the Schulz Bank Seamount.
It was a pleasure.
Yeah, it's been beautiful.
We've seen so many amazing sceneries and animals. But we will be back. I mean, we have other plans as well for tomorrow. You will see, but
Or the day after.
I think we will be back because we've only scratched the surface of the Schulz Bank Seamount.
Yes, we have.
Yeah. So more to come.
Yeah.
Bye.
Bye bye.
Bye.